Good morning, afternoon, or evening, wherever you might be joining from, and welcome to Dive 553. We are back down 2.5 kilometers below the surface of the ocean at the East Pacific Rise. We're back at the Fava Flow Suburbs. Today we're going to be checking out experiments 2 and 3 again, but we're also hoping to be collecting water samples as well as some more biological sampling as time allows. So welcome, and as always, if you have any questions, feel free to type those in the chat either on YouTube or Facebook if you're watching live. You can also follow along even when you can't watch us with the hashtag #VentUnderworld over at Twitter.
Okay, so we are at the experimental site. Can you do a frame grab here? Yeah, on this one?
This is the video one.
Yeah.
Okay.
The other one you already did, right? Grabs?
Yeah.
We're also just touched down at one of the student experiment sites. So of course we have the choice between meat and vegetative matter up there, but we were also just looking at a little satchel of beans. There's another site, another deployment of this experiment over at a venting site, an active venting site. As you can see, the squat lobsters do not seem to appreciate the potato.
Or the beans.
Or the beans, yeah.
Or the chickpeas.
Yeah, no, they just really don't want any of that, huh?
And in the middle here we have the fat as well, and they don't seem that attracted to fat.
Yeah.
Up top is meat, and this has shrunk quite a bit. As you can see, it's pulling little bits off. And at the bottom is liver, which seems pretty attractive here. But chickpeas, beans, and potatoes do not seem to attract them very much.
No.
So the other experiment is right here on the left hand side. These are different kinds of beans, and then a little packet here to see if they will sprout underwater. There's no sign of sprouting yet. Here we have similar experiment on board to compare to this little bag, and we check on the experiment on board every day. Some of them are sprouting already, so we'll get back to you on this. Now I'm gonna move to the active site with the same experiment. It's at waypoint 16.
For me to take it.
Yes. I will. I will. There are some right here, but for now we're just moving to the experiment. And there'll be some around. So all the stuff you see there on the edges.
Yeah, this is all barnacles. Oh, that would be a good shot.
Yep.
Is that one of the potential biological samples today is barnacles, or?
No, it's just we needed to illustrate. But the these guys are very interesting. They're always at the periphery of hydrothermal vents, and we found different species in different areas of the world.
Mm-hmm.
These tend to not be very abundant sometimes, and sometimes they're just everywhere. So we are at the second experimental site. I'm just gonna do a quick zoom onto the barnacles. No need to move. I get something a little better than that.
Yeah, you got some active feeding going on there.
Yeah, there's the tail of a fish. The barnacles are actively feeding. You see
That's nice.
the cirri extended, and once in a while they retract them to bring the food back to the mouth inside this little shell part. You can do frame grabs here if you like.
Yeah. Could you maybe do like a more zoom out?
Zoom out. Yep.
Yeah, just to take that a little bit better. Yeah.
All set?
Yeah.
Cool. Gonna check on the experiments now. Fish. So for a little later, this limpets are something we're interested in, so we'll be trying to suction them later. Okay, experiments. So it seems some riftia have fallen onto the experiment, but that's okay. Up and to the right was some liver, and this disappeared pretty quickly at the beginning of the deployment. Middle to the right is the fat, untouched. Lower down to the right is the meat, doesn't seem to have been touched either. We have here a large crab coming in. So if it fed on that thing. Anyway, the meat then seems untouched except for the liver and for the potato, untouched. The beans don't seem touched either. And there's the chickpeas behind the riftias, and can't really see what's happening there.
Try to lift the riftia.
No, I mean, I don't want to risk messing up the experiment. I think it's good. We know that the liver is gone, so there is not much to see there. I'm going to zoom onto the beans in a little baggie there. No obvious sign of sprouting either. There is bacterial growth, though you can see the white stain on the mesh bag.
Mm.
Okay. So I'm gonna have a wider view of both experiments for a frame grab if we haven't done so yet.
Cool. Thank
Okay, so here we're gonna try to suction some of the big limpets. That will be the first sample of the day. And we go to a coarse jar, and that would be 6 or 7.
As folks are trickling in, once again we've returned down to the bottom at East Pacific Rise. Specifically, we are going to be visiting Fava Flow Suburbs again today. This
Okay, we're
is
at
live
6.
footage from the remotely
We're in
operated
6.
vehicle
Okay,
Sebastian.
perfect, thank you.
If you're watching along on YouTube or Facebook, you can type questions in the chat. We'll try and answer those as we can. We're stopped here for a while, and then we're going to be visiting and checking back up on experiments 2 and 3, the mesh box experiments. Yesterday we collected up mesh box 1, or the experiment number 1, which yesterday was quite successful in a lot of ways. They also were able to collect a fantastic rock sample, and in the collection of that sample they were able to see that the gap, the empty space below the rocky crust there, actually did have organisms living in it, which was a large goal of this expedition, was to try to find evidence of that. So quite good. But thank you for joining us for today's dive. We're just getting started, and we have lots to do.
Well, I think the whole thing fell out of its holster, so the nozzle you're grabbing, it's not really supposed to be there. Just
Grab a soft with the tips.
yeah. Just keep the blade in the soft part of the handle and just get what you can get, pull it out of there, we'll get a better grab. Not sure how you will. Yeah, I doubt it's gonna stay. There's nothing set it on. I'm gonna pick it up and put it on the board quivers or something.
So this instrument here is the suction sampler. If you've not watched our streams before, we do have many ways of collecting samples, including this particular hose, which sort of vacuums up samples into small containers on the side of the vehicle.
Back here.
Don't really know until you release it what it's going to do. Yeah.
There's some sort of cover head here. Yeah, so
Okay, so this is rubber T5. Site.
I guess it's the first.
Where is it going?
Sorry. Four, suction sample, 1, 2,
Six.
6 goes into 6. This is like a fine match.
It's coarse.
Course.
Yeah.
I'm not sure. I haven't caught up with what sort of samples came up on the big rock itself from yesterday, but the 20 or so worms that we collected into the bio box made it up to the surface just fine, and the cooler also doing all right. The rock was quite heavy as anticipated, but I know it was looked at pretty thoroughly last night, and I anticipate they'll continue doing work with that rock for a while.
There you go. You gotta get through it there. No, don't. Straight on ones are terrible. You want to pitch over. Just lay down in it.
So we've been taking a number of different samples throughout this cruise. Been taking water samples, which we can be looking for microbes in that water, or looking at the chemical composition in the water, that sort of thing. The suction sampler has been used for collecting bacterial mats, but also more macrofauna such as the limpets. And let's see what exactly we're vacuuming up here.
No, the ones, see, there's some, I don't know if you see me, Alex, on this. Yep.
Yeah.
So these guys, and there's more in the back there, so we'll start with these.
I had your assumption.
Excellent. These are crustaceous.
So not getting the worms themselves with the suction sampler, but
This one would like as well.
getting the things that tend to be on them, a little bit off of them.
These guys there.
So there are little limpets that we
talked about.
keep seeing on these tube worms.
Should I come next to you, or you good?
Sorry, what?
So yeah, this
Yeah,
could be one, but then these guys down there.
Okay. Yeah, just keep in mind he's super focused on the one he's.
Yeah, I know.
He's probably not seeing you. But,
Yeah,
as
that's
soon
why I
as-
was offered to move over there to, if need be.
Little pink
Yes,
scale worm
these,
there as well.
If they can, if they will come.
But that scale worm is kind of
Or are we expected to?
they
battened
will-
down a bit.
Yeah, they will hold on to the rug pretty nicely, so you can brush them with the hose. Try to see if they come off, and if they don't from here, then we'll move on to another spot.
That's kind of what's.
He's in that little groove.
Yeah.
Yeah, he's pretty fastened down in there.
Okay, if they're clinging pretty hard here, so just need to keep track of time to not get too distracted. Let's try to get these guys then, if you can, on the little piece of rock there.
Sure.
Yeah, it's still going.
Oh, it's still attached.
Oh wow. So these are Lepidodrillus crustatus, but there were some nodopeltas, but they hold on to the rock so hard they're not coming off. We'll try up there as well.
Yeah.
And so far we haven't seen Max's art.
Sorry, you haven't seen Max's out?
Yeah, maybe it's fallen behind the rift gas there. We'll see.
But it's not there where it was?
It's there, we just haven't looked at it.
Ah, okay.
You saw it.
Yeah, we were
Okay.
flying over it.
Just
Okay.
like 1 meter up from here. Exactly.
Okay.
I send you the pictures.
Great, excellent.
Yeah, so just
Just
check.
now?
Yeah,
Okay,
just check if you I just looked like there was like some art.
yeah.
So.
I think you got one.
Where is it? It's not this. Yeah, it's the one from
Yeah.
Okay. There are a few more over there. You see the yellow dots? That is reachable from where you are? Looks like it.
Up on the rocks.
Yeah, so you have some there
Yeah, okay, good.
on the rock surface.
Oh, further out.
Is it too far off?
Not necessarily. Let me porch out.
Oh, porching out, sorry.
I'll give you some more slack. That should be over.
Yeah. So maybe the surface may be a little easier to work with. Yeah, these are the ones we want. If they will come.
So you can see those little limpets all over the tube worm there. We've been trying to get some of these
That's
organisms,
pre-dissected.
but many of them like to fasten themselves down to the rocks pretty well. So it can be a bit tricky to get them.
Yeah, they always go in those little tiny grooves.
Yeah,
to protect
yeah.
themselves. It makes it really difficult to get.
Yep. I realize that. And we always want the ones that are not easily accessible, right?
What about the ones on the Riftia right below it?
Different species.
Oh, really?
Yeah, that's the problem. So, see, these have little nubbins on them.
Okay.
These are smooth, different shape as well. Maybe on top there, maybe easier.
Comparing the different limpets that are found on both the worms and the rocks.
There's none on the mussels at all that we could just grab the mussel.
No, not this species again. That's specifically in this kind of environment.
Okay.
So. So this one is okay as well. I like if it's easier for you right there. These two as well. Oh, in that area. Of course, it's in a hole again. Okay, maybe a little further to left, we'll see. That looks pretty good. Ah, damn it. All is gone except the limpets. We may be able to get better ones on the left-hand side there. Easier ones to get. I don't know. To the left side of the screen, yeah. You see all the yellow dots, the yellow spots there. These are all the limpets that are interesting. This one seems to be just hanging. There's a good chance of getting that one. This up there. Still there.
Yeah, so as limpets, they should be able to move around, but they're also
It's
infamously
something
strong,
closer to us.
yeah,
It's
fastening
it's
themselves
venting
down to
just
the
a little
rocks.
lower.
Yep.
We're just tapping the.
Yeah. So here you see the yellow as well.
Is there any in this area?
There was one up there, if that is accessible.
Let me ram in.
Okay. So right there. V1. Thank. Yeah. Yeah.
Okay, that came off.
Excellent. I haven't seen it coming in yet, but some fuzz is coming, so that's fine.
Yep, boom. Just
You got it? Cool. Okay, what time is it? Just 2 or 3 more minutes of this and then we'll have to give up on it. Just trying to look for...
I got us a little closer, so if you want
You
to
do?
try those ones that were further away again, we could give it one more shot.
That's, you can try in that pile there and there. There may be some we don't easily see, but
The method of how these animals travel around from vent to vent is one of the points of interest of
you
this
tried that. It may be difficult.
expedition. So one idea was that they were perhaps traveling below the seabed here itself,
It's okay.
being taken in by the
Let's
venting fluid,
do this for now.
being taken in where the venting fluid is being pulled in at recharge sites below the surface, and then when that water gets pushed back up at a new vent, it would distribute those animals right there. And with yesterday's expedition, or yesterday's cruise, we were able to get some good evidence that that is a possible means of relocation for these species.
Hey.
Out. You can also at the end, because you go there, if you have something left, you have to suck up among the algae leads to it at the end and take a little bit more time.
Okay.
You know,
I suppose not relocation, but spread. You know, where the offspring in their larval forms would move,
Okay. Yep.
and how new vent sites can be colonized by these species.
Here after this. Okay.
Yeah.
That's an easy one. Crab
When
spice.
you do this, you
Yeah.
did already this, no? Also,
Yeah,
you
but
tried,
we haven't looked at Max's art yet. It's all for
yeah,
her.
that you do for a few minutes.
Yeah.
And
Yeah.
then you
And
get
do
this
crab
here,
spa
you
and
go
then the
here
main
right, and
part of
then you
it.
will see how long this all
Metal
will
back
take.
out.
Yeah.
So we don't have
And if you
space to
cannot go, because this is longer, then you
Yeah.
just go,
Straight to
Here.
Bishop's Spire.
yeah, take time for sampling here. If
Yeah.
time
Yeah.
allows and request, just play with.
Yeah.
Because I have the feeling
Yeah.
this here,
Yeah.
probably not.
Probably not.
And,
Or do
so-
we trigger them?
Yeah, she will tell you.
Okay,
If
she
I
will
see
do.
her,
Okay,
also because
cool.
she did not know yesterday.
Thank you, guys.
Now this is in the way.
The lid? Yeah, all of it.
Can
You
reach
think
out
we-
and grab that lid and just tip it down?
So that's one of the questions. We have a question in the chat there about whether or not, or how would those organisms, or can they, withstand the temperatures involved in that process of being taken down through a recharge site, being heated, and coming back up where the vents are. And obviously they can survive the temperature where that venting water is coming out of the ground. We've measured that at
Cool.
at our
Thank
experiment
you.
sites at about 20 C at those diffuse vents. It gets much hotter at something like a chimney or a black smoker. But yeah, I'm sure that they'll want to measure the temperature of the water from where they pulled the rock out yesterday, because that opened up a nice little void space underneath. And there were organisms living in that space.
I think Stefan what he likes is that you see the frame, so basically the
Yeah.
frame base, and then just zoom out so it's not too much zoomed out, just a little bit centered in the middle and go a little bigger, right? Something like this.
I'm trying to see all the bits.
This here, I think it uses that to put it together. One sample down over.
Okay. Okay. Next stop is a crab spa.
Which number? 6.
6 so far.
Thank you.
Oh, got a worm on the loose.
Oh, so we're still floating.
Yeah, probably dislodged it with the thrusters.
Oh, poor guy.
Yeah. There are a few others around.
There's a question in the chat about what sort of study's been done into the oxygen requirements of like the larval worms. You know, if we're talking about them traveling below the ground, that water there would be low oxygen. But I mean, yesterday when we popped the rock up, there's things living there, so
Yep.
they must make do. But I'm not sure about any studies specifically into that oxygen.
On larval forms, it's not done a whole lot.
Yeah.
We know that they consume oxygen, of course, but it's difficult to work on them to have enough and measure consumption of oxygen on things that are that tiny.
Yeah.
However, for adult forms, we know they are able to actually regulate their oxygen consumption down to fairly low concentrations. And that's very good ability for these guys. Typically, the crabs or some of the scale worms we have worked on, they're able to work down to about 10% oxygen saturation, which is pretty amazing. Shallow water species could not do that. So,
Yeah.
and a key player in there is the presence of hemoglobin. And these hemoglobins have high affinities for oxygen for the worms and scale worms. And for the crabs, it's hemocyanin, a copper containing blood pigment that gives it a blue color.
Yeah. Yeah.
So we're heading to Crab Spa. It should be on our right, I think. Oh.
Not right here.
From this angle, I can't recognize it. Is it? I think we just.
That's the same offset as the rest of them for today.
Okay.
I thought that's what we did last time. We came over here and there's
Yeah.
this little cup. And that's
Yeah,
what
that should be it, right? Well, you guys know this place a lot better than me and how to approach it, so.
I'm not positive.
Okay. There's a small spire in the back. That's Bishop Spire, okay. This is not it. It's beautiful, but this is not Crab Spa. No.
Let me spin back around. Might be that one behind me there.
Yeah, up there, I think, isn't it? Maybe. Ah, no, it's not it. Unless...
I didn't know.
should be something that is open like this, visible to us. This is not it. Zum was jetzt, Stefan? Or not?
I'll go get it.
Oh, thank you. Is that thing straight ahead there? Is that it? It opens up on the top somewhere. Looking for crab spa, but
On the other side,
yeah, that's what I thought.
yeah,
Other
just
side.
just here.
That up. Okay. Thanks.
I mean, not that this doesn't look nice, but it's not.
That's not what we're looking for.
It's not the opening.
I knew asking you that. It will be like-
No. It's still yeah. I got even confused when we arrived the first day. You know, I was yeah. So this person I already thought it was first spot, but it's not. Yeah, you can actually very nice sign is the weight stacks here. We
Yeah.
deployed large volume pumps right here,
All right.
and then put the hose here.
Okay, here we are.
Yeah, that's perfect.
Wonderful.
Great.
Thank you.
Yeah, of course. Oh, you're doing this first? Okay, yeah.
Oh, we've done with the students experiment, and we're
Yeah.
doing a crab spa first, and then we're gonna
Yeah,
go
that
to
makes
the
sense. actually,
experimental
yeah.
site.
Right.
So
do
here
some
we-
nice limpet and snail detection, yeah.
Exactly. Here we will want to collect limpets and thank you, Stefan.
Yeah, sure.
Limpets and snails, so suction samples as well. So just so that you guys are not.
You want to grab that before we go in and land down?
Look at the crab in there, hanging in there. And Zavina wanted some from here as well, correct, Monica? Yeah, she wants fine, super fine, and coarse for the limpets. For the snails, if we didn't get enough.
Then we have only, okay, yeah, good.
Yeah, she wanted a whole set here because it's essentially the same as
Then we
subsurface.
take a whole set up there. And we have more than a whole set up there because we have 3 fine, 3 middle, 2 coarse. So this would be the last course we can sample. because-
Yeah, so if we see plenty of snails coming into the
Yeah, then
fine,
we
then
cannot-
we'll not spend the course here.
Yeah, don't spend the course here because
Yep,
we might spend it up there.
yep.
And then we have 2 fine and ultra fine is good.
Okay, cool.
That we fill all our suction containers with this, no?
Yep,
Yeah.
yep.
Okay.
One fine, one super fine.
Yeah, now and then we have left super fine, fine and coarse for up there.
Yep.
Okay.
For up there, you mean at the Fava flows, right?
Yeah.
Right.
Okay.
Wait, because then she also needs one from Bishop's Fire.
No, then I cannot take this because the 3 we need to keep for the other one. So you used 1 now, no?
I used one, yes. If it's, but she wanted a sample.
Yeah, but this is less important than the one up there.
Okay.
Let me see. I talk with her.
Yeah.
What we can use.
So we'll be working that hole the whole time. So I'll end up there a little bit.
little tight. Yeah, that's what. See.
So all the little round beads you see everywhere are the snails we're going for here, and depending on what Zabina wants, we also may use one of the finer mesh.
Huh.
You okay?
Yeah, just looking at something.
Okay.
What in the world is that? You see that in the document? Yeah, oh, I see what you're thinking. I know exactly what you're thinking. Nice and stable and should be good in there.
We do
Oh
nothing
my
here,
God, exactly.
and we keep
Just
the rest as bishop
Stay
style
right
for the
there.
albumin leads, if we have whatever we have
So
left.
for the fine and super fine.
For everything, because,
Find out
yeah.
which.
So we don't
basically,
do we don't
have-
do ions here.
No, we do nothing here because we need. We have like fine. This is coarse. This is another coarse.
Yes.
No?
Yeah.
So we need, let's say, click for our side,
Yeah.
and then we need click for.
Bishop
Yeah, and
spire.
then we do more for the bishop
Can we
spire.
do one fine here then maybe?
No, leave it, because maybe just leave it, then we like nothing.
So
here.
we're not doing
No.
anything here? Okay, for a
No,
future dive then?
because- Yeah, we keep this.
Or we can add it to the other sample we had from the student experiment site for Ian.
No, don't
No?
keep doing the
Don't?
same. No,
Okay.
no, then he has to wait.
So no
for a while.
section sample of this one?
Nope.
Okay.
Change of plan, sorry. So we're going to Fava Flows, waypoint 13.
Okay, that's what we're under a section sample here, we're gonna move to the boxes. After the section sample? you-
No, sorry, we're not doing sessions. We realized we will likely not have enough suction jars if we do the suction sampler here. So we're gonna first do the priorities for this dive, and then if we have time, come back here or on another dive maybe.
So we're going up. Back up.
213, correct.
Alright, so come up high and over there. I'm gonna try to bend that mount out so we can actually read it. You have your arm. You don't move your fly away. Using the orders. Okay. Better. Of course, the way I think it's not perfect.
I don't recall if we took temperature here previously, but NSF has crab spa listed as about a 25C diffuse vent.
Weird. Okay, we also need to get this closed. So
Okay.
real quick. Oh. Really? That's not inside the brand. Blades.
That's fine.
Blade's good. Yeah, blade's good. Oscar's looking at all. Maybe I've been too far and it's not gripping anymore.
No, I don't think for some reason it just didn't want to go down.
Oh, I mean,
I'm gonna get
Yeah,
it off.
touching
I'm
the
gonna
chin.
get it way off.
oh, wow, snake.
Was Crab Spa impacted by the eruption in 2005 or
Crab Spa was discovered after the
after?
eruption.
Okay.
So I know this story a lot less than Stefan does, but basically after the eruption there were a few sites that were active, and one of them was Crab Spa, and ever since it's been visited every time we dive here to make sure it's still active. And there's some Stefan in particular work on bacteria from there, microbes from there. And he collects samples every time he visits to follow the evolution of the populations of microbes from there.
Okay. Coming up on just about an hour since we've started our stream today. For anyone who might have joined us in that time, we are back down 2.5 kilometers below the surface at East Pacific Rise. We were just at an event known as Crab Spa, and we are kind of just going to be traveling around Fava Flow Suburbs again today. Probably check back in on Meshbox 2 and 3 after we collected number 1 yesterday. But we're also taking the opportunity today to collect water samples as well as biological samples as our time and space allows for.
Okay, so we need to check on the 2 remaining experiments.
Oh, yeah, there's only 2 now. I was like, oh shoot. Where's the other one?
Wow.
And then number 2.
Mm-hmm.
I don't even think one was really the most bacterial growth on it either.
It was not. It seems like two had the most, just from my point of view.
Yeah.
Is the most temperature that I think.
Yeah. There was so much yesterday just in that box.
These guys still there. They're always
I love them.
hanging out.
I think they're my favorite thing that we see regularly down here. Honestly, I don't know, something about them catches me. The little flocks of because they're just sitting there. They're riding the thermals almost. You know, it's kind of cool. These little swarms of amphipods that we see above some of the venting areas.
Actually, site 3 looked pretty like it's caught up to 2.
I think Site 3 might be surpassing it, but it was younger, right? So
It is,
2 really took off, but 3 seems like it's maybe the crab's definitely picking at it.
Well, good. Thank you, Alex.
So these are sites 2 and 3, experimental sites 2 and 3. This is 2 here on the left, and 3 was the one on the right there. We were just discussing the bacterial growth that we're seeing on these boxes. So it's sort of that white fluff. That's filling the holes in the plexi acrylic.
Bacterial growth there a whole lot.
So when we collected box number 1 yesterday, as they lifted that up off the seafloor, it was filled with this bacteria, and it looked like we were in a snowstorm for a hot moment there. They were, even with as much that came out once we lifted the box up, they were still able to stick the suction sampler up underneath the edge of the box there and just collect a bunch of that bacteria. And then once they popped that rock up and opened up the void space underneath, there was a lot of bacteria down there as well. So certainly a lot of bacteria.
We can't leave it on a jar.
We did a course jar, so we had 6.
We only did 6 so far. Is that your question?
Thank you. I moved it off of 6.
Oh,
So when you're
just
done
make
with the
sure,
jar,
yes, you don't accidentally.
yeah, it'll just, it has the ability to siphon it out. So
Got it.
I moved it just in preparation. The good thing is, like, I like to turn the flow to just barely on, so it's still pulling, and then I rotate it. As soon as it rotates off, I turn it off. You know what I mean? That way it's still holding anything that's in there in the shower.
That makes sense. Right, so before you turn it completely off. Got it.
Okay. From here, can I see the other one, or do we need to move?
Prediction going on there. Gravity.
Good. Don't need to move. That's a lot of bacterial matter. Alright, I'm gonna have a wider angle for you,
The crab is eating.
Andre.
Yeah.
Wow.
So how many days? 4 days, I think.
Yeah.
Oh,
It's
is it also
kinda
like the- what's
a little garden,
stuff?
yeah. You know?
Yeah, it's purple as well, right?
Yeah. Which is actually good.
Unless it means it's so anoxic in there that another kind of bacteria is growing.
But that's like already, oh shit. Oh, so.
That's It's like a European seat all the
Yeah.
time.
We just switched to Dutch and nobody's understanding what I'm saying.
No. Really?
Scale worm.
Little scale worm.
Yeah.
I thought I was like two.
Wow, but that's massive coverage.
Yeah. The top. Frame
Oh,
grab.
even the big one is filled there.
Yeah.
And that's like 4 days down there now. Or not 3, but 3 or 4 days.
4, I think.
4, yeah.
Wow.
Whew.
Yikes.
Yeah.
It's,
This needs to come up tomorrow.
Nah, but we don't change the sequence of the,
Oh,
the
it's
same fall.
the third?
This is the
This
third
is
one.
third.
Yes, that's the third one.
Ooh, never mind. Sorry.
But you know what's the...
Sorry.
I go here. It's also like here and here.
It's just, I think it's just detaching because the conditions behind it are no longer right to
Yeah.
let fluid through and allow probably this bacteria to
Yeah.
grow.
Cool.
Yep.
Maybe we should think about changing. I don't know. Changing the sequence. I don't
No, I mean, we can't understand it. It's perfectly fine. I thought this one was number 2.
Yeah, no, it's
and that's
number
why it
3.
was so overgrown. Nope, it is not. Okay. Can we change our viewpoint? So from the other side, please.
So just before we take off, I think there's a crab eating the gills of a worm that I'd like a shot of. The crab
Is there?
over this way. Or past that. This little guy there's
know. In the
going.
background.
Ah,
Left,
yeah.
spot.
So it's squat lobster.
Yeah.
Grabbed a piece of rift, yeah.
Mhm.
So it looks.
Oh, nice. Real lights.
Hmm.
Look at the shimmering at the same
That's also
time.
very nice.
Good catch. Okay.
Just carry it.
These kind of big boxes, yeah. Just
Yeah.
I send you more pictures.
Which means the rest of it, so you are. This way is going to take out the Vina.
Yeah, the whole. It's a bit less on this side here. I think.
Good? Yep. I'm stealing the camera. Grab.
So again, you can see the bacterial growth here on this side.
Cheesecake.
This is box 3 now on the left, and it'll be 2 on the right.
There's some there as well. Where the flow is coming out on the silicon.
Did you see the snail-like creature on the box?
It is a little bit. The one right there.
On the corner.
This one or higher up?
To the right, on the corner higher up.
The right. Sorry, I'm gonna zoom out to get my bearings. Oh, this one.
there.
I mean, there's a worm below it too, but
Yeah. Yep. Oh. Ah, that's a little bit. Okay. Yeah, they seem to appreciate bacterial mats.
Yeah, I think that worm was there the other day too. Yeah, just like right on the corner. Yeah.
It's even growing on the, on the frame.
I'm afraid, yes. Everywhere. Here it's possible. Rolling around. Oh, it's inside the plastic there, right?
Yeah.
Because it broke a bit, you know,
Yeah.
and then it can go between the
Yeah.
plastic protection and the stuck.
It's a good place for hiding. Okay, so that was box number 3. Deployed 4 days ago. And box number 2 was 2 days earlier. The purple coloring of the mesh is everywhere now. So we have a little bit of torn silicon here, but still working at the bottom. Still tight. Good. Trying to see if there's flow from the top, but it's hard to tell. I see some, but it's coming from the face. Oh, go for it. Okay. Snails grazing some more. Okay, so we're gonna go to the site where mesh box 1 was. So there's a big hole in the ground now. Should be easy to find. I'm gonna zoom out to give you a wider view.
Was it off this direction or am I disoriented?
No, I think you're doing it right. Maybe turn your head to the left. Just get over.
Wasn't it?
Somebody steal our hole?
Doubtful. How'd you find a missing hole? By the way.
Also, if you see the bar, it's going to be really close. Because we left the bar right there, I think. The red bar.
Should be like
Go for
somewhere
it.
here in this area.
Right there. It's right there.
All right, so we are
Thank you,
approaching the site of experiment number 1, which was recovered yesterday, and the pilots very, very successfully pried up a rock. A large slab came up, exposing the open area underneath, which now the amphipods have decided is a great little spot to sit and ride the currents.
I'm really curious what happens here in the next coming years.
Yeah. No, that'll be a thing for sure. Is now come check out the new hole every once in a while and see. It's got bacteria now, but at some point it'll have worms.
We'll see what we can do about that.
Can I steal a camera from you then? Thank you.
Just sit down on that rope and see how we can see the money that
I mean, you could hook the loop. Then someone's gonna have to get it.
Look it back on the biologs like it was originally planned, where we put it on the back of that one. Get it out of our way.
Also make a
Hmm?
highlight from like, because we don't have anything clear.
Yeah. Just want to see the ripples on this one? Okay, so do you want a highlight video or
No, just a frame.
Just
that we
frame
have,
grab,
like,
yeah.
a nice picture from the hole.
so we'll take this zoom and then I'll zoom out a little more.
Take another one.
Yeah, please. Thank you. Trying to see where the most of the flow is coming from. It looks like it. I was wondering if down below us was also a good spot. We'll measure temperature in different places anyway before we start sampling. So, I'll zoom in a little more. There's a bit of a rubble there. More of the stalactites.
It's a very good structure.
There's another layer underneath.
Yeah.
Let me see two worms there.
Yeah. Can you zoom in?
I think I'm all in.
Because
We'll
the
see.
or is it the rock that has fallen in? Probably.
This is this crust of this
Mm.
iron sulfide orange. This made to green anyway.
So here's one hanging.
Yeah. See the stalactites up there again. Okay,
This
so
corner is belonging to us. This is the other side of what.
Yeah.
oh, the rock.
Yeah, this is the other side.
And here's also one hanging like towards the
Yeah.
subsurface. And you can see here on top the
This is a stained worm here.
grass. No,
Red thing.
no, that's,
That's
no,
just
that's
the
a
plume.
pollen.
That's the plume of a. Oh, no, that's a
No.
scale worm.
It's a scale one.
And this is not.
This, yes.
Yeah,
It's
this is
called
scale worm, yeah.
normal color. Yeah. Okay, so we want to measure temperature, find a place where we have good flow, and we'll sample water from there
Okay.
with the temperature wand, so into the small bag.
You can
We
see
get
that the
closer
stain.
to that or just grab it and see?
Let's push out and see if I can do what we're talking about with the hook, get it out of our view, and then have that out of the
This is low bed water. Net below water. This is low bed and low bed. This is full of water.
It's not for me to grab it off the tank and we'll push out water.
Now that I'm looking at it, put that pin in the direction.
Nice. So we'll start with the corner closest to us, probably. Just make sure it's. And we'll need to read the temperatures.
Hey Alex, can you make the temperature of
I
that?
got you.
Be excited to see what this is.
Probably should have brought the channel for this.
It's such a large area, it's diffusing.
Yeah, almost background temperature.
So you're not
Yeah,
here.
okay. At the
Okay.
same time,
Makes sense.
it's not like sort of.
Well, yeah, let me know.
Okay.
Sure.
We'll move into that corner, please. Pretty good. Let's see what we get there. It's a little warmer. Not by much, 4 degrees C. Okay, I think going to that corner will be probably the warmest, and if it is, that's where we will be doing the water sampling as well.
Is it possible to stick it in a bit?
Sorry?
Is it possible to stick it in, like a few centimeters into the hole?
Oh, like under the hole?
Yes.
You can try.
In the same area? What you're saying?
The same, the where we were just before?
No, like
Really?
the one, the hot one, like on
Okay,
one.
the warmer one. Yeah, what we think is warmer. So in that corner, chill. It's the attack of the amphipods. Yeah, we're under the overhang there. Oh, nice. We're already at 12 degrees, 13. Increasing a little bit, we'll be back, I think. Still going down. Probably. So, it's very local temperatures. 7.6 here. We saw higher, so we'll try to find higher again. But I think this is where we will be working. And so this time we're gonna be sampling water into a small bag for an hour and a half. So is that a place where you can hold for that long? Yeah.
Okay, let's torch out a little bit, give me, so we're not so much tension on this cable. Alright. Feel a little better by now.
Okay.
Float in your face though.
Well, they're just getting back so you can see the temperatures.
Okay.
Cool.
Yeah, well,
Good here. We went up to 13 degrees Celsius at some point. We're now at 8.
And is that on the seaway?
it's UVSVX, I got it right here.
Oh,
So you just let me know when you're ready and I'll start it.
Good for you. Do you want to find higher?
It was originally 20 with 20 marks.
I
You know, when you remove the rock, because it's
So maybe try to find a little higher temperature if you can, Carl.
Okay.
So, but it's not surprising we're lower now because we're so open to the outside. It's not capped.
Can I see the tip of the jaw?
yes, absolutely.
Thank you.
Yeah.
I
Oh,
think
to
a little to
what?
the left.
Oh, that's good. Stay there for a while. See, 9. Even further left, maybe, or deeper into the overhang. It's funny. We go deeper, it's colder.
Yeah, I think there's a little current that's drawing some salt water in.
Yeah. Oh,
Just really
refreshing.
trying to find the hot spot.
Yeah, trying to find a little higher than that if you can. Here was pretty good earlier. Let's try that. It's going up. 10. That's a good spot there. 15, 16, 18.
20! Stay here. 22.
Stay.
Let's stay there.
23.
I won't touch it.
24. Stay.
All right.
This is good.
Very good. Interesting worms as well. Okay, so let's start it, please, and it will be.
So we got a timer ready?
I'm going for it. Yeah.
They started now.
Okay. 16:11.
I don't like this. I don't like this.
So this is sample line
Because it's like, and it's gonna rock.
water sample on the tiny
That's it.
Yeah.
Because there's no fire.
I mean, that's good.
You're good for another cigarette. That's that.
All right, so if you've been along on some of this before, we are collecting water, not into the regular bag actually. I was hoping we'd see the other one, but there's a different bag.
I'm sorry. In a while.
So the other bag I just showed you is the one we've been using, but there is a separate bag for this particular probe. But we'll have to turn the main camera to look at that, so we might do that in a little bit.
Yeah. Yeah, so. Forward and it's.
Yes, so it was 90 then.
90.
Also the 90 is
90 is real Fava.
Oh, yeah, I watched it carefully.
Real Fava inside of it. I don't know, I cannot see the.
Yeah.
Okay.
See the tube going into
I'm
the
going to give you a
vein.
bit of finesse here.
It's on the first set.
Yeah.
Yeah, because you just put that it's too late.
You want to see the metal sheet running good, but not the.
Yeah.
So the first test today was 95.
Yes.
Okay, and then we have 96. This was not the second. This is not the second one.
96 is the second.
So this is the one we're doing now.
Yeah, the water sample the tiny bag.
Maximum temperature was 24.9.
I locked. Let me see.
That's it, sit up here.
Yeah, 24. Yes.
24.9. Little bag filling for. We need to go 6 liters is 1.5 hours.
Yeah.
Stefan,
I don't know.
I need a little bit more than 1.5 hours. 6 liters is 1.5 hours.
Yeah.
I need,
Another 15 minutes?
Yeah, another
One more liter?
yeah.
145, guys. Is it okay if I play around with the camera, or do you need to keep it on there?
Nope, do what you want.
Try to keep the people busy online.
So this,
Okay.
85 screw back. Yeah, 86 suction truffle. Yeah, 7 suction truffle. 88 sponge and big truck. 89 rock sample the small bag back. 90 rocks. Yeah, 91 tubes collection. Jesse. Ah, that I forgot. Okay.
Any idea what's going on with those little sections? It almost looks like bamboo, you know how they make sections?
Oh.
You can see it on the smaller dark tuber right
So
there.
this one, right? So this is Oasis. Oasis has different growth pattern. It will grow and just flare out, and then grow again straight and flare out.
That's what's doing it.
Yeah. Whereas Riftia is continuous growth. Sometimes you see little flares as well for on like the one on the right hand side. Yeah. Temperature is dropping a little bit. I'm gonna go back to the placement just to see if we've moved for some reason. Oh, it looks alright. Looks alright. We know it's gonna go up and down anyway. Just making sure we didn't move drastically.
So the pump is on.
The pump is on, yes.
Can you take a look at the bag?
Right now it's gonna be very flat. And
Yeah,
we
yeah.
just started.
Yeah. That's true.
I'll look at it in a
Yeah.
few minutes.
toasty water.
She just should have tossed her egg in that hole overnight. Let's see what do it, huh? Took long enough, maybe.
Just like room temp.
CJ, feeling with his bacteria once in a while?
Yeah. Mm-hmm. Can you zoom it?
It's zoomed all the way in
Oh,
already.
it was already. Okay.
Yeah, pretty skilled work.
I'm wondering if these sometimes look a little bit like these at the bites on the snail that have the spotting happening.
Yeah.
That literally means the back.
But I'm not sure.
The Fava Margarita?
Yeah, maybe. But,
Oh, out of the shell. Yeah. Yeah. And meant like you put a full leg in it now. That kind of works. Just
Cave
and
system,
yeah, sounds good. I would just make sure your elbow over there. It's such a waste.
Go
No, that would be my bad. Yeah, that'd be gross. But yeah, it was just the milk. And I was like, this kind of it was just spread it. That makes sense.
Yeah, yesterday we did, after we popped the cork up on this rock here and had the opening, we did actually scope around down inside there with the manipulator camera for a little bit.
Their last leg reached over the back.
Yeah.
They can scratch their own back.
Yeah.
Only with the last leg.
That's so jealous. That's so cool.
Yeah, they do that in something as spot shrimp at home. Their front little arms are actually this, and they're full of, it looks like this. Just like this whole 50 joints, so it's like super full.
Go gadget.
Yeah, and so they'll bring that up and they'll like reach back and be like, bring them and scratch and stuff. It's so weird that they back. But it's like ultra joint. It's like flex ups. And one is more. It's a little tiny pincer. It's just a little bit bigger.
Underworld, aber das ist Connection auch nicht so super. Das ist natürlich der Underworld. Visible cave seems to be visible.
Oh, went to 25.3.
And Underworld.
Cool.
Cave, caves, Underworld. And Underworld caves.
We saw the landing of a sea cucumber there.
He's something of a pilot himself.
It's a little far to get good lighting there.
Yeah, so when the double solid box doesn't have the best ceiling, so when they were having issues, yeah, they mentioned that they had like a cooler idea. That actually wasn't the first cooler. There's a little bit more rigid one, and we tried to modify it to fit, but it was just too cold.
Well, that was the Pelican one.
Yeah,
The white one.
realized
That's good to know. We were talking about why like just a half for this, and it was like Pelican, but it's too tall. Well, they make smaller ones. They do for sure.
So the cooler that's on the front porch, that was provided by the scientists as a potential solution to getting the worms and other samples up through the warmer waters. Although yesterday we put 21 worms in the smaller bio box, and apparently they got up here great. But our larger bio box, it just doesn't close as nicely. So we just want to try and protect those samples where we can, and the cooler turned out to be a great solution for what we're looking for. But the bio box, the small bio box yesterday also got that job done. Because of course we had to use the cooler for the rock, which you lifted the rock. How much would you guess? What do you think that rock was? Because I don't think they weighed it officially. But.
It's quite heavy.
4 people carrying it.
Let's say 60, 70 pounds maybe?
Yeah, okay. Yeah, we were thinking it might be something like 30 kilos. Yeah, which is about that. So you think more even?
More because it's more.
Wow.
The 40 kilos I can carry
Because
with
we-
Sabine, but we could not
We
carry
could
it.
to do like for sure more than
Well, you were carrying the water and the
also
rock.
water, but also the rock when you lifted it out. Okay?
Yeah.
The water.
We're guessing the rock maybe 30 kilos and the water 40, so the total box was like 50
No, the
kilos.
whole box was heavy because we needed like I don't know. With the water, I guess like 60, 70.
It was impressive,
Keeping
yeah.
you busy with the
logging.
And
Yeah.
we
As
used to
well.
carry like buckets for 30 liters.
Yeah.
This you can carry alone, but this was not possible.
So we're thinking the rock may be as much as like 40 kilos. I mean, it's a solid slab of the basalt. It was very solid.
Does it look like it's coming up?
Well, we haven't looked yet because it hasn't really ran so slow, you know. Trigger 15, 20 minutes is probably asking to take a look at it. So I put the one in on the upper left side, and I didn't find the hot water for obvious reasons until I got it to the roof of the inner cavern. That's where we're seeing this. So if anything goes wrong, you've got to put it back. That's where I found it. Yeah, just taking a sample. I moved the bungee from the cooler from that front to on the back of Hydrobox 3 just because the monkey system was really in the camera view. So.
Yeah, we need to use a real pipe to get that out again. That'd be good. A hook on the back of all those hinges.
Do you want to see or do you still want your 6 minutes off?
I'm fine, you can put it off. Not too loud. James, something like that. Yours is 6. That's far.
Small bays, sometimes with ventals, with no bays.
It's not as light now, eh? I threw another block of light on it.
Oh yeah, it flies really nice today. Yeah, I noticed it the first time I switched it on with chill.
Nice and smooth now.
Oh yeah, it's good. Yesterday wasn't
So there's one of those swarms or flocks or murmurations of the amphipods back there. You can see them kind of all streaming up in the water.
We're just over there.
We're up to 25.5.
Excellent. That's what we like.
Aber es steht eben sowas,
More upstairs. There's a bit of kind of like, I've got my back. It's better. I mean, when I first did it, it still hurts. I can tolerate it. But between you gave me stuff I got to tell me, like really, really hot on my back, but that makes the rest of me really cold. No. So it's working. Look how pretty.
Give it to my dad.
From graphic.
Doesn't last that long.
It's, I mean, it's like you get snaps anyway, so so purely.
If you're just joining us, we are currently pumping some water up from beneath the ground here, collecting a sample, and the temperature that you see on your screen is also the temperature at the tip of the probe where we're taking that sample. So on yesterday's dive, we were at this site. This was experimental site number 1, and we picked up the experiment, and then we went to collect some sediment, some of the rock under the experiment, and the pilots were actually able to pry up a large section of that rock, and it exposed this sort of void underneath it where there was clearly growth and organisms down there. So scientists were very excited about that, as we were trying to find evidence of fauna living below the surface here, or at least making their way through the vent fluids as they run below the surface. But today we have checked on experiments 2 and 3, although we're not collecting either of those 2 today. We're going to be collecting number 2 tomorrow, I believe. But today we're just sampling. Water samples, geological samples, and biological samples, I believe, are all on the table today. So that's what we'll be doing. Again, this is at East Pacific Rise, and we're near Fava Flow.
Crabs always like probes.
I guess it's curiosity and feistiness.
Take that thing out of the cave. Especially last night.
If I go any further down, it'll probably pitch us that way.
Right, stable, sweet.
Yeah. anything, we would go downward, go forward more, and see if we can push in that way. In front of us.
Aber sozusagen, wenn es das so zeichnet, wir sind nicht, also unser Projekt war dann praktisch über das raus, so.
Oh, the tell you, pressure test
Die zeigen sich das dann so, dann Projekt, das sind über gar nichts wissen. Genau, das ist jetzt falsch, aber das ist wirklich.
The only disadvantage now is when it's being raised, it will corrode.
Und der Schnitt ist es so, weil der Schnitt ist so umgekehrt, genau. Also, suchen wir irgendwie, obwohl das gescheit auszusuchen, damit man das irgendwie so ist.
I'm gonna have a quick look at the bag to see if it's filling up nicely.
Go ahead.
So as I mentioned, it's a different bag from the one you might be most familiar with us pumping water into. So we're going to take a quick look at that. It's mounted in front of the other bag, but behind the camera that looks at that other bag. So we have to use our main science camera here to view that one. You can see it back there underneath the mesh.
It's been filling for 20 minutes now, so it's probably a liter or a liter and a half in there, so it's not very visible yet. Just wanted to have a quick look.
But this is not the bad.
For visual, this is 1.9 liters.
What is? Yeah. Think you have enough coffee in there? It's
There's
water?
a lot of- yeah. If you drink a lot of coffee, then I'll rehydrate with the water.
What time is the pump now running?
Sorry, say it again?
What time is the pump running?
You still have an hour and 22 minutes.
So,
Trivia? Next time.
You guys are too good at this for me.
We have a question about how long ago was the lava flow active? Not that long ago at all. 2005, 2006 is when the last eruption in this area came through, which is just fascinating. So the slab of rock we got, like 18-year-old rock basically.
It's like the legs now.
There was a question about basalt versus andesite. Here we're pretty sure it's basalt. There's no subduction anywhere nearby to actually allow silica to get into the magma chamber. So I've seen andesite in the Western Pacific, but here is all basalt.
When we flipped that rock up yesterday, there were some really interesting, like little stalactite sort of situation on the underside.
I'm putting one onto the screen right now.
There we go. So do we think that's just kind of when it was molten, it was dripping down a bit, or?
Yes.
Yeah.
And because they're so skinny, it's probably in a gas phase, so probably water had completely, and gases from the magma had filled the chamber, and it was just dripping into these small ones. and then later water came back in.
Okay, fascinating. And did we probe down at all into the bottom of the void? Is the bottom of the void there rock, or do you think that's built up with like just bacterial sediment at this point?
There's rock rubble,
Okay.
but we also have the white-ish, sandy-looking things there that probably are bacterial mats as well.
Yeah.
And we're gonna sample these later
Oh wow.
for
Oh hey,
people on board. Hey, the crab's here.
crab's hanging out in
Another
25
one.
degrees C there.
2 crabs.
Bye. I mean, we've seen them eating the bacteria along the box, so this
Yeah.
is, you know, we have opened up a little grocer for them down here.
And they're close to their limit there.
Yeah.
At this depth, probably their upper temperature limit is around 25 C for normal physiological function.
Yeah,
They can probably withstand a little higher than that for short periods of time, but
yeah.
also the higher the temperature, the lower the oxygen concentration. So
Yeah.
They're pretty good at extracting oxygen from a hypoxic environment.
Yeah. Of course, you know, all these vent creatures, they really do always skirt that line of being just too close. Like,
Yeah.
I mean, when we were over at Crab Spa, you mentioned there's that crab that was just tucked right down in it. So,
Yep, absolutely.
It's investigate early development in giant reports. From embryo to
and a few folks were noting, and I think Jill and I had actually been discussing, we'll be interested to see, you know, if this site is then revisited over like the next year or the year after that, what will it look like now that this hole has been opened? What sort of stuff will show up?
Crack it.
My prediction will be that tube worms will be colonizing the edge of this
Yeah.
hole.
Yeah.
I will put this to the test because I'll be back here in January,
Oh,
so
very
I'll
cool.
be able to check this out.
Yeah, no, I'd love to know, because yeah, like you said, I mean, all around the edge then gets sort of colonized, but it's got this little bare spot in the middle still. Who knows? Or if that'll fill in. Yeah.
I think it's gonna stay open like this, but and the growth will only happen where enough flow is coming
Sure,
out. So
sure.
in the center, it's gonna be too much like seawater. Probably
Yeah.
not much will happen, and then on the edges, probably a lot. Tube worms and mussels may settle there, and as they are there, they will divert the flow slowly towards
Yeah.
the center, and they may eventually colonize the center if they plug it enough and allow more flow to come to the edge.
That was something I was going to ask, right? Is like with like new forest development, new forest growth, right? You have trees that eventually sort of block out the sun that not as successful trees need. And those successful trees, or those less successful trees, then die back. So with the worms, do we see or do we anticipate the worms sort of diverting enough flow with their growth that other worms that had been there get cut off?
So it's possible. I'm gonna put on the camera here.
Right.
So you have Riftia, and down here you have Oasis,a the smaller one with the flanges. Oasis,a deals a lot better with harsher conditions.
Okay.
They tend to be in higher flow areas. Once the tube worms settle there, they will plug a little bit of the hole.
Sure.
But in the succession we have observed here over the years is that later mussels will come in.
Okay.
As they come in, they will settle on the tube worms and kind of smother the tube worms
Sure.
a little bit, if you will.
Yeah, yeah.
And by consuming and diverting the flow at the base of the tube worms, eventually there will not be enough reaching the plume of the tube worms. So what we have seen here is typically high flow. Another species of tube worm yet settles there first.
Yeah.
Then Riftias and Oasisias move in, then mussels move in, and you end up with mussels only in that area.
Yeah.
And that takes place over probably 5 or 6 years.
I love that there's like succession regimes, again, kind of like forest development. It's very,
Yes.
very cool like that. Yeah.
Yeah. And that was discovered because here we're lucky enough to visit shortly after an eruption.
Sure.
So we've been able to follow the whole succession there.
If I would zoom in on that pink critter below
That,
yeah.
that's a small alvinella worm, probably got disturbed and got out of its tube. It's a palvinella.
Okay. So I have a procedural question for you. Also,
Go for it.
So what we witnessed yesterday was very affirming in the hypothesis that was presented as to, you know, where the worms grow from underneath, and we picked up the large slab to say that. What would be the next process in verifying that? Would it be just studying exactly what comparisons you have from, like, what would be the next step? Saying, okay, well, it looks like what we thought might be happening is happening. How would you go about, say, proving it or validating that? With that?
Huh, that's a good question. So yesterday the worms that were observed were most of them attached to the stalactites, and we only saw the tubes, meaning at some point the conditions were right for them to grow under there. So likely larvae came from elsewhere, got channeled through subsurface and settled there, and the conditions were right, but they were no longer right when we picked the rock. It was probably right close to fractures we have between these slabs. My point of view is, if you really want to test that, maybe it's a bigger, heavier colonization device you want to put in there.
Okay.
This is great for a relatively short time, but probably you want to put something that will sit on top of the crack and kind of channel the flow out. So like Monica said earlier, larvae cannot swim upstream. They're tiny. They cannot fight the current that's coming out there. So maybe one way would be to put like a, I don't know, concrete chimney top, if you will, and under which this flow can be channeled, and then try to see if after maybe 6 months something came and settled underneath it. Could be an option there, but it's not easy. All experimentation in the deep sea is difficult. This is probably the most complex setup I've ever heard of, trying to understand what was going on there. Experimental ecology in the deep sea is very rare so far. We're trying to do some more, but it's expensive, technically difficult, and sometimes difficult to reproduce as well. So it's difficult then to share the information and publish this information because people want more replicates. So far it's been a little over half an hour, still have an hour and 10 minutes to go. You may have gotten thicker. So that bag can contain up to 10 liters, so going to 7 liters will not fill it to the brim. I'm not worried it's going to blow up or anything. It does look thicker right now. And heavier.
Especially look at that corner right there.
Yeah. But I've noticed during other dives they just can make a dome once in a while, and it's hard to know if it's full or not. Same with the blue bag. If you only see it from top, it looks like it's full already, and right now it's completely flat and empty. But we need to keep an eye on this one to see if the creases are disappearing or not.
If you notice, because just with the current and the slight flow in RV, we're getting a little bit of that edge. Is that okay still?
I think it is, yeah. We're still measuring around 20 degrees. If it's dropping much below that, we'll probably have to reposition. Still going in and out once in a while, so for now it's good. Also, you guys online may have noticed that there are some rusty areas as well. So rust stains here, for instance. Iron oxidation like this cannot happen if there is hydrothermal fluid coming out. So basically here you have a little bit of rubble. It's possible you get some warming through just conduction, but there is no fluid flow here coming out and reduced flow. So the flow contains iron sulfide, and you would not be able to oxidize iron in these conditions. So that could here be an outlet to slightly warmer water, but just heated and not chemically modified, or it could even be an inlet into the subsurface. Quick look at the bag again to see it's filling up nicely.
Just take a look outside. We do have a little bit of weather coming in. Doesn't look too bad, but overcast and maybe a little rain occasionally can affect the connection a bit. So if we get a little sporadic here or there, definitely have some patience with us, and we'll try to get that strengthened back up for you.
The creases are still here, so I think we're good for now. Still have an hour and 5 minutes to fill that bag up, so it's been 40 minutes so far. And there's a cloud of amphipods at the bottom of the ocean.
Two of them.
Two of them, indeed.
Yeah.
Gnats.
Yeah.
So Alex seems
Yeah.
to like them very
I
much.
like
to me,
them.
To me, they remind me like clouds of mosquitoes. So I don't.
I mean, it's
Yeah.
not like I'm a stranger to mosquitoes where I live, right? Like, yeah, but I don't know. It makes me kind of think of like the murmurations, like the flocking patterns that you'll get in starlings and that sort of thing, where
Yeah.
it's just it's a cloud of life, right? Like
Yeah, it's mesmerizing. That's for sure.
yeah. And I like it as well because they seem to be kind of like they sort of ride the flows a little bit there, right? They're like riding thermals or where the flowing vent fluids are coming up. So,
Ah.
yeah. Perhaps not, the most glamorous of the creatures down here, but just as charismatic. I
They're
think.
very cool.
Yeah.
And they're pretty tough.
They have one fan.
Yeah, they got a fan in me. That's fine. That's fine.
And you're right. There was a blob to the right that has shifted completely to the left now.
Yeah.
But there's a counter current coming back in.
Yeah. A time lapse would be fun on them. Just see kind of how it like moves around. But certainly, I get the gnat and mosquito comparison as well, for sure.
So right below that is mesh box number 3, the one that was deployed the latest, and as you can see, it's very, very colonized by bacteria. These will be picked up in 3 days from now. The first one to be picked up of these 2 is going to be mesh box number 2 right here, and that will happen tomorrow. Check a little bit on the wand again to make sure it's not moved too much. Good. It's still where we want it to be, still measuring 23 degrees right now. Look at them. They left us.
I remember we were off the coast of Oregon one trip. It was right after lunch and it was really dark in the control room. And I'm flying around. Then I look over. Everybody's asleep. So I look over to the chief scientist to make a comment. I look over and she's asleep. There's only 4 people in the room. I was the only one that was awake. It's like, all right.
The smaller tube worms here, the little white ones.
Yeah.
Do we know what do we know much about them or?
So these are serpulids,
Okay.
a family of polychaetes. these filter feeds. So what you see there are the crowns, and with the crowns they can aggregate particles that they eventually bring to their mouth and eat them. They are more abundant close to hydrothermal vents because there's a lot of suspension in the water. However, they are likely not endemic of hydrothermal vents. As you can see, they are in colder areas. Also, the green color you see in there is due to their respiratory pigment, which is called chlorochlorine. It's a hard one. It's very close to hemoglobin.
Okay.
Just the heme group that binds the oxygen is slightly different. There's a little modification there, and it just appears green in the body.
And then, I don't know, I've seen these a couple times, like the little bulbs that come out of some of them.
Yep.
What's that structure there? Do we know?
So it's a modified head structure that when they pull into their tube,
Yeah.
just closes the tube.
Oh, okay.
Protection. Like close the door behind you. The door is attached to the filament. Earlier we could see some that were, so maybe this guy here will be able to see it closed.
Almost like the like a perculum on a snail or something.
Absolutely.
Okay.
Absolutely. See, I think this one just below the rock
Yeah.
is with a perculum close there.
Okay.
So this one's just leaning on the ground right now. But if it retracted, it will close it. And you can see it's a little brown to the very tip.
Right.
It's because it's also rich in keratin.
Okay.
Same compound that makes our hair or our nails. And this keratin is just a little harder. So if something tries to grab it from the top there, it will just resist being pulled out.
Sure. Yeah.
And here you can see the tube worms are actually leaning and trying to stay as close as they can to the crack where some fluid is coming out. So if there was a much higher flow, these riftias will be standing up, and here they just try to stay close to the ground and to the crack where the fluid with sulfide is coming out.
Like a plant kind of following the sun. If it,
Absolutely.
Yeah.
Same principle here.
Sure.
Chemosynthesis requires sulfide and oxygen, and CO2, of course. And the sun, if you will, of chemosynthesis is reduced compounds that are found in the vent fluid, and here it's sulfide, so they stay close to the origin, the source of that sulfide.
Mm-hmm.
For the ones we saw in the background earlier and stand upright, a lot more. The big bushes there.
Yeah.
These guys, the flow is so high they can actually stand upright and still get enough sulfide from the water. And as for the amphipods, we don't really know why they swarm like this. We see some of these guys sometimes on the tube worms,
Yeah.
but the swarming is a little unusual. So maybe there's enough particles in the water as well that they can actually gather them and eat and feed from them.
Yeah. No, I was curious if they were kind of just grabbing particles as they go by.
I think it's very possible.
Mm-hmm.
But these guys always make swarms. I'm not an amphipod person, so I'm not 100% sure that the ones that crawl on riftias are also Ventilla. But this one, Ventilla, is often making swarms like this.
Mm-hmm. Nice squat lobster just hanging out there.
Quick look at the bag, see how well we're doing. Definitely thicker and heavier. Doing pretty good. Still have 50 and some minutes to go.
How many? 50.
50, yeah. It was an hour and 45 minutes total. It's been only 50 minutes so far. The wand is still measuring between 23 and 24 degrees Celsius, so it's still where we want it.
So obviously we have a lot of the venting around here. Do you know much about the recharge sites that are feeding these vents, or?
No, that's one of the big questions. So
All right, yeah.
there are two things to consider here. For making the hot fluid, that's probably happening hundreds of meters, if not kilometers away.
Sure.
That seeps down into the crust, gets close to the magma chamber where it's modified by the contact with the hot rock. And that's what we see coming out as the black smoke. Obviously, it comes out at 350 degrees C. Nothing can live in there. So there is another thing happening, though, and that's what we're looking at right now. Basically, before it reaches the surface, there could be some dilution with seawater.
Right.
That seawater will enter much closer to the vent.
Yeah.
And then you have diffuse flow happening, and that is what gives you the prime conditions for the vent ecosystems to develop.
Yeah.
And so that happens probably much more closely, and that could be just a few meters away.
Yeah.
It's possible. But the recharge of the hydrothermal fluid itself,
Yeah.
hundreds or maybe kilometers away.
Yeah. So then, you know, with this idea that there's organisms traveling through the fluids, I mean, how far away hypothetically could things come from and make that process and not enter when it's still too hot, but you know.
That's a good question.
Yeah.
I mean, it could just be a few meters away.
Right. Yeah.
So here we are in some sort of a dome, and we saw it's broken on the side.
Yeah. Yeah.
So that just the fluid coming out could be at the same time sucking some water in
Yeah, just a
from
little
the
bit.
sides.
Yeah.
So that could be only like 10 meters away.
Okay.
Still pretty significant.
Sure. Yeah.
Because larvae being driven by this
Yeah.
will be funneled through and just will end up in the right place. And that's the whole
Yeah,
point of this proposal. I wouldn't expect it to be much more greater than this distance-wise.
Right.
I wouldn't expect it to be more than, say, 100 meters.
So the organism's not getting drawn in like the prime recharge site because they still have to go through the massive heating at that point, but they're getting kind of drawn in at like these secondary sites.
Absolutely, that produce the diffuse flow we're seeing
yeah.
here. So
Fascinating.
there's a little bit of the pure fluid that
yeah.
comes out mixed in underwater,
Sure.
under the surface, and then it's variable.
Sure.
Condition-wise, the hot fluid besides the temperature, of course,
Yeah.
is also completely anoxic, and
Sure.
all the animals we know require oxygen
Yeah.
at least at some part during their life. So we need to have not too much of the hot fluid in there as well for that
Yeah.
reason. And then the other conditions are quite challenging in there as well.
Yeah.
High CO2, low pH,
Yeah.
some heavy metals in there, but these guys are equipped to handle these kind of challenges.
Sure.
So yeah, a little pinch of hot fluid is okay. Like more than 50% probably would not be okay for the organisms there.
Yeah.
I try to have a look at the bottom of that opening here, but shimmering water always makes it difficult to
See if I can get through that a little bit.
just maybe aiming it in the center will help. Oh, nice. Thanks, Chris. Getting there. Maybe a little more. Yeah. Cool. So I see a few snails down there. It's obviously layered. You can see another hole coming from under this broken rock there. Nice. Cool. It's been an hour now. Another 45 minutes.
Yeah, diffuse flow is like give me an opportunity to practice my shutter speed, focus, try to like see through weird stuff.
Shimmering always makes it difficult. So observation throughout the years have shown us that here, about every 10 years, we reset the whole hydrothermal ecosystem there, roughly every 10 years. It happens at this pace on fast spreading centers, but in the world we have much slower spreading centers. So on the mid-Atlantic ridge, probably a lot less often than this. There's a site where the Europeans have been working a lot. It's called Lucky Strike, and it is something that's probably been active and relatively stable for 5,000 years. In the West Pacific, lots of unknowns there, but probably also fairly long-lived. Somewhere in between the two values I gave you will be probably a good estimate. So maybe the recovery after disturbance is also not as good for the sites that are a lot more stable. I just saw a fish that was either munching on something or it's not visible from this camera, unfortunately. Never mind. You can see the tail flipping. So there's a diversity of stability and response to disturbance around the world. So what we've learned here, which is probably one of the best known sites so far, probably does not apply elsewhere in the world. So we have to keep observing and see, especially after disturbance, how fast they recover, essentially, in different places in the world. We still need to learn a whole lot more before we can even think about doing anything there with a human influence. Like deep sea mining, that was what I was aiming at there.
Sure. Yeah. Nice crab wandering here. I've noticed this stance with a lot of these crab, with the last set of legs there up in the air, sort of
Yep.
head handstand situation.
It is typical of the family. The family is Bethodidae. We find them at hydrothermal vents only, and the leg is permanently, this last set of legs is permanently set upright like this.
Okay.
It's probably because they're pretty good on chimney walls and tube worms. So they hang there with the last pair of legs attached, well, grabbing something in the back.
Sure.
And they can actually face outwards.
So the legs are kind of just permanently positioned to be holding onto a wall behind them.
Exactly.
Almost. Okay, yeah.
Exactly.
Very
Hang
cool.
there. If the legs were lower, then it would probably be facing too much down
Sure.
when it hangs. So that would allow the species there to hang more upright in the water. Another thing you'll notice for these crabs is they have little, like tiny little eyes or no eyes at all.
Sure.
Can you autofocus on this or? Thank you. Sorry. Wonderful. Thank you, Chris. So as you can see, the eyes are tiny. You see the eyebrow, if you will, and little white eyes. So they're reduced. They have no pigments to perceive light, and probably non-functional. And in some species in the same family, the eye is essentially absent.
Yeah. Yeah, we'd spoken about this with the shrimp, right? Like some have eyes, some don't. But you were saying, obviously, when they're larval and they're planktonic, they may be up high enough where light is a thing.
Yep.
But obviously down here, much less so.
Exactly. So the study the species. I can't remember which one. And they saw the expression of proteins in the ice that are consistent with the ability to see for larval forms, but not for adults.
Oh, fascinating.
Those options.
Yes.
Yeah.
It's remarkable. I can't see yet yesterday when we were getting worms. As soon as something landed near them, it was like, bam.
Yeah. They're pretty good at feeling vibrations, and this is how they detect potential preys. Quick look at the bag again, getting to the point where I want to make sure it's not blowing up.
Yeah, that's,
Yeah.
you were that right there.
We went up to 26 degrees C in the water right now.
Well, not doing anything with it, but just, I mean, if we have to do that, valve open, it's gonna be right there.
It's behind the oil, so just need to be careful.
Yeah. Where did that come from?
It's the one from the valve. That's what it is. Tied up with the coil.
Run that up?
Yeah.
Oh. Let's do this just for a second.
Yeah, go for it.
All good?
So yeah, the only one that we consider is the lower one.
Right, I just wouldn't have a problem. So you should bring it in and just shoot.
Yeah, that's more about like grabbing it though.
Okay.
It's a little small for you there, Alex.
That's all right. That's all right.
John and Nash. Just watch the movement. See coordinated with others.
Just keeping you busy.
The worst.
Nice. So other things there, you see little red tufts at the bottom of this crack. These are the gills from a small alvinellid worm, Paravinella. These and Riftia have hemoglobins in their blood. That's what's providing them with this red color that you can see. This hemoglobin is very much like human hemoglobin in that there's a heme group. It's the same. It's an iron center, but annelid hemoglobins are a lot larger than human hemoglobins. Human hemoglobins are made of 4 globin chains. The worm hemoglobins you have here are 144 chains in one molecule. It's just a huge edifice there. So big that they are nearly the size of some viruses. Just retracted.
Since we were talking about.
So say it again.
You could just sense we were talking about it. It's like,
A little shy.
yeah.
We're just coming out though.
Is there any oligochaete species described here in EPR? No.
Yes.
Yes?
There's one.
There's one.
Yeah.
It's like a leech or?
It's a leech, yeah.
Okay.
Not big at all, but there's one.
Okay. So what it parasitizes?
It's been found on crabs,
Crabs. Okay.
but maybe just we captured them and then they were just moving around with the crab.
Yeah.
But they could also parasitize the fish, for instance.
Okay. But it's easier to catch a crab than a fish.
That, and I mean, so for us, it's
Yeah.
easier to catch a crab, for sure.
Yeah.
But the type of leeches that are there look like the typical type for fish.
Okay.
So we think it's probably parasitizing. Great.
Yeah.
Parasites on fish.
Yeah. Okay. And how do you catch a fish? You have like a special way of catching fishes? Or
Yeah, we take a net and we.
Net?
Yeah.
Okay.
At
And
this depth, the way that we would do it is we would identify which one we wanted, and we would chase it.
Okay.
Because the deeper and the colder and whatever the metabolism for the fish isn't super long. You know what I mean? Their metabolism doesn't give them a lot of energy for a long period of time.
Yep.
Okay.
So we chase them around, wear them out, and then use a net,
Okay.
wrap them up, and then bile box. Sometimes if we're lucky, we'll be able to unwrap them inside the bile box and get the net out and get more. But
Okay.
that was, we did it quite a bit off the western coast of Australia in 2020.
Okay.
So
Otherwise, other submersibles sometimes use their suction sampler.
yeah, we through this
Well,
whole
like
event.
full blast.
Yeah.
And you can suck them in like that.
We were catching fish like this big for
Really?
nets.
Okay.
Uh,
Like butterfly chasing, right?
Pretty much. Yeah, yeah. One pilot with the arm with the net just waiting, the other pilot just flying around. A lot of
It must be
communication
fun.
back and forth. It became fun. At first, it was very frustrating, and then once we kind of got our plan and our rhythm down, it was fun.
Moving to make sure we're still in place. Temperatures dropping a little bit, but that's not unexpected.
It's going up again. Oh,
It's going up again. That's not bad. We are still in the right place, so no worries. And now we're back. We still have half an hour left.
So I think one of the issues. What's up?
yeah. That's fine. Yeah. Good luck.
What were you talking about?
Good luck.
The launch.
Oh, we're doing good. Yeah. Yeah, he looks like he's so pretty.
We had a little more light here earlier. Is it possible to?
Yeah.
Thank you. It's filling up nicely. Happy about that. Like I said, another half an hour. Oh, 27 minutes.
Oh, really?
209
Oh, for your
Inception.
crowbar with your bar with the car attached.
So this is the place where the telescope should be?
No,
No, oh, okay.
that's actually right next to the controls, all the triggers for the Niskins. The mass bike was back on the right. Essentially the same as this one, but across from it.
Oh yeah.
Yeah. Almost there. Some more. Okay. Thank you. Great.
We need to hook a little power drive up to that dial.
I think the highlight of my day that I listened to that thing. The first time I did it, was you sitting there and it was just like, and that noise.
Well, yeah, there was one where you were going for like 30 seconds at least.
Yeah. I mean, there's a time or two I'll go the wrong direction. And it takes me like 15, 20 seconds to realize it. Oh,
Sure.
oh, gotta go back.
This one's a little different. It's kind of raised up a bit.
Yeah. So sometimes they just crawl, sort of, or they build their tube
Yeah.
along the surface, and sometimes they just get a little more erect.
Sure.
You know, other places at least is just take better advantage of the current flow. But here, I don't know.
Yeah.
likely as well.
Oh, we got some of the silicon
Yeah.
still there. Yeah, it almost needs like a little marker in the middle of the hole because on our way over we were like, where'd it go? It just blends in with the seafloor.
Is there any evolutionary explanation why, for example, circulites have like a keratin tube and reef that has a chitin tube?
So for circulates, it's calcareous tube.
Calcareous tube,
Yeah.
okay.
No, not that I know of. Trying to think phylogeny right now. Ciboglenids, which is the family to which Rictia belongs, all have chitin tubes.
Yeah.
Calcareous tubes are not that common for polychaetes. It's only in circulites.
Okay.
And among the circulites now you have spirorbids that were considered different family before, so also calcareous tubes. Yeah, that's all I know. That is also circulate. Just doesn't live attached. Yes, of course they do.
Yeah, it's interesting. Why they tubes they're made of different polymers or proteins or carbohydrates.
A lot of annelids just use their mucus to agglomerate small particles to make tubes. That's a lot more common.
Okay.
Some of them make a silk-like substance with which they line their tubes. Also, trying to consolidate the sediment.
Mm-hmm.
Yeah,
I'm assuming these are harder than rift jet tubes.
Oh yeah, a lot harder.
Rift jet tubes are quite squishy.
Yeah.
Yeah.
Riftia, Ositia, Tevnia, Virginia all have squishy tubes.
Yeah.
Same type of tubes for cold seep species such as Lamellibranchia,
Parascarpia.
Parascarpia as well.
Yeah.
I wonder how warm the fluid is. You should get the probe all the way under the hole. As far as you can reach.
RV bridge.
Not too warm, yes.
Go ahead, Bridge.
And it's going to come 10 degrees to starboard.
That's fine.
So what is the logistic there? The vessel always need to be
Yeah.
close to the
Baby
RV.
steps.
Close to the RV, and they also need to keep their heading so that we're not moving too much.
Okay. So every time the vessel is moving, it needs to adequate to the RV position or the other way around?
So the vessel's going to move to a position that is easiest for them to maintain that position
Okay.
due to wind, currents, sea state, whatever.
Mm-hmm.
And so we'll set up the vessel to begin with
Yeah.
that in mind. And then as the day goes by, they might adjust it here or there, and they'll let us know so that if it's a move that's too much for us in the position that we're in, we'll do both the heading change and a boat positioning change.
Okay.
And it's just to make sure that our umbilical is safe, and they're not going to put it in a position where, like he was saying, like it's going to put too much pull on us or whatever, or moving on top of us or anything like that. Like for example, if he needed to go, like 30 degrees one way or the other, then if he's going to port, then I'd probably move him over here
Okay.
so that our launch point is still kind of a direct spot from where we are to where they are.
Mhm.
Again, if he was going starboard, I'd move over that side, things like that. Now, if he needed to go like 180, then obviously he'd go down here. It would take about
And also currently
35
for the
minutes.
VSAT satellite that we're streaming with, we don't want to go any further to starboard or we're going to lose connection.
secondary priority. Connection we're most worried about is that the other one.
It's the big one. I told that was like, well, just go to port. But yeah, I guess you could shuffle forward a little bit because we don't have any cliffside or anything we're dealing with right now. Because we
I mean,
deal with that.
It's fine for us if you need to move for you, rather than we can have them do that.
Yeah, well, he knows, so.
Okay.
If we get to that point, then just address it. That means the other internet is fast for all of us. Or
Good.
you on the email.
Just
that's
cheat.
handling it.
Sorry, Chris, I'm moving
Oh,
around
no,
a
no,
lot.
I'll just cheat. Here we go.
Thank you.
Yeah, I can do that whenever it's far away. The focus, if it's so close, I gotta do it manually because
Okay.
it's confused.
I mean,
It's like, ooh, look at that.
Yeah,
Oh, look at that.
yeah, unfortunately. No, GVO fluids. Yeah.
You should buy me a camper.
Evan just wants to buy a van. I was like, well, that's 10 grand. You can buy a 12 inch for 139, a trailer for 500.
I don't know. Are you allowed to weld it on yourself?
But you can't sleep in the back while you. If it's,
Oh, fine, it's small one.
if it's a van though, she can sleep in the back while you drive.
Evan's a princess. She only does about it. A fair bit.
That's not a DC, right? The day room.
So it's like, so you gotta say, I guess, on the side, the band strings, got a fair on band strings. Here's one of our good friends, my home used to be, what do you call it? Like a, behind us, chicken house.
Yeah.
She lives in Raleigh, beautiful, that.
What's going on?
$10,000 more than it was last year.
Oh, What is?
So You just mentioned something.
Ah. Okay.
It's 34K. But it's $10,000 more than it was last year.
I don't think this is inflation. There's more.
I think it just got popular, so they're like, ooh.
Yeah, probably. So on the Riftia plume there, you can see all the gills, filament, and you can see also longer filaments, and these are sensory filaments. So if they're touched by a predator or an ROV, they can retard in their tube as a reaction to that. Silver crab down there as well. If they can get a piece of that, they will go for it. So the pink lips sort of at the tip, actually the field cartilaginous, basically they play the role of an operculum on the top of the tube. In Riftia they're just called opturaculum. The central part of the gill is pretty stiff as well and will allow the gill to be fully extended in the water. And then once in a while, when it needs to, it retracts inside the tube, and you only have the pink part that is exposed. It's also, there's a little bit of keratin in there as well. That's what you see as a yellow tinge to it. So it's just protection there to not be exposed too much when they are inside the tube and close to the opening of the tube. "13 minutes left. Almost there."
Almost there.
As a species, how old are these worms? Like, how long have they been around?
The origin of the lineage, like the vent ones at least, it's about 100 million years old.
Wow.
There are different species in the world, and so that origin is probably between 65 and 100 million years old. But the group, Ciboglinid, is older than that, and you have some species that also live at cold seeps, and then you have other closely related ones that are simpler, let's put it this way, and still symbiotic with sulfide oxidizing bacteria at cold seeps. They used to be called frenulates before. And again, close to that, you have Ocedax, the bone-eating worm. It's the same family as well.
The what eating?
Bone-eating.
Okay.
So you find them on whale carcasses.
Whale bones, I
Yeah,
know
yeah.
them.
Yeah.
Never cut one open.
They smell awful.
Oh, yeah.
That's
But that's not.
So,
okay.
the whale bones actually contain a lot of lipids, and these worms will just dig into the bone essentially, and they have symbiotic bacteria, but these bacteria are able to degrade fat rather than use sulfide as an energy source. But the whole family is specialized in symbiosis.
How do they bore through the bone?
They produce acid from their posterior end. Doesn't have to be very concentrated, very slowly just goes into and just makes a little alveol and just until it gets in contact with the fat, and then it just, yeah. And there was a paper by someone who worked on fossils, and he found in the bone of a fossil whale, probably, the same type of structures you would expect with these worms. Doesn't mean, well, he suspects that this type of worms already existed at the time and were eating the bones the same way. "10 minutes to our sampling." Let me check on the bag real quick. I don't think it will make any big difference right now, but
Were you talking about also that? Or?
yes.
Yeah, they currently published a genome paper. I think it was like maybe 2 months ago. and they found really interesting things there. Like the whole metabolism behind this bone degradation is really different than ocedax then, or the genes that degrade bone and all. It's like really different from ocedax to other worms that are close related to it. So it's really interesting work.
Yeah, he's encountered a whale fall.
I think some people are able actually to cultivate ocedax.
Yeah.
Yeah, they just need some bones and then they can create the right conditions for it.
Yeah.
Yeah, they can study. Yeah.
So they actually deployed some, I think that was cow bones in North Sea at only 50 meters, and they got some that settled down there.
Wow.
Then you can keep them in the aquarium.
Yeah. That's crazy. We saw them off of San Diego. I think it's Rosebud. It's the whale that washed up, so they sank it with chains.
Okay.
I think that was it Scripps that sank it?
That was the Gerger screws.
Yeah. Yeah. It was the last time Jesse was out there.
Yeah, I had to fly that for Julie Peterson.
Yeah, we were doing the
3D,
photogrammetry.
Yeah. I remember that. Yeah.
Crazy 4 a.m. piloting.
Yeah.
That group.
What if we pan up slightly on the city?
That's it? Yeah.
On the sit. Yeah.
Probably 6-7 minutes left.
Okay.
Yeah, I have 8 minutes left total.
It was close.
I was like,
8 minutes.
You don't want to miss the exciting end to that sampling, right?
Yeah, I had to brush back over.
okay. It's almost done. How was lunch?
It was alright.
Okay.
Not the most exciting. This is chicken pie stuff.
Yeah. I looked at that. I was thinking comfort food.
Yeah, digging into that hot sauce that Alex Engel brought.
I saw it sitting over there. I didn't have any.
No. I got.
We can't handle milk, and can't handle hot sauce.
No, it's not good. I can't wait for spicy yet. What's that?
It's tricky. Somebody said that you were eating that with dark chocolate because you can't handle the regular chocolate either.
Yeah, 90, 85, 90 something. It's just cold what he's in.
It's probably a shorter list of stuff you could actually eat up.
Can we get the temperature back, or?
John,
Just as
John, can we get the temperature over there?
Sorry, just to make sure it's not drifting. I mean, it looks pretty. Placement still looks very good.
We can do it right here as well. So it's in the one. Alright.
Oh, that's
Alright, but it is back over there.
Realize that.
okay. Yeah, it's all cooler than before, but that's the way it is.
His is bigger, ours is quicker.
What you guys want? You want it here or you want?
I don't care. Whoever wants to go eat first.
You want to eat?
You can go eat.
Sure.
Yeah.
All right. Should go and see what salad stuff's available.
So, yeah. Oh, by the way, there's a cable.
It fluctuates a lot, the temperature.
Yeah.
Is taken down. I'm gonna look at that. I'm gonna look back real fast.
Yeah, sure.
Yeah, it's right here. It's not an issue like of going anywhere. It's just whenever if we deal with this, we'll have to be aware of it.
Yeah, so for the timing, I can show you there.
Oh, he never zipped that back up.
Now it's fine.
The
Yeah,
we-
we took this
coded to
off
video.
so that you could
Okay,
do the
that's
work to it.
Show you.
I guess you got to put back on, but never too tight.
So that's the thing. Good thing when I was first with you, I was thinking ahead.
Yeah.
When we designed this years ago, we're like, you know what? They're not gonna tie a cable up right here. That's not what that.
So you make big bucks, Chris.
I made the big bucks.
So when these will need to be turned on, no further than this, essentially. It's already on when it's there. It doesn't have to be all the way back. But Tihomir will be here.
We got you.
close to the end.
So,
A little under 5 minutes left.
we're sampling right now, are we, in the big bag?
It's small bag.
Small bag, that's Capri Sun.
Yep. And we put probably about 7 liters of water in there. So it's taken an hour and 45 minutes.
Why are you taking so much? Just because it's a cool spot, or?
Just because Monica wants to make sure we're gonna get something there if we have issues with the blue bag again.
That's a good call.
Yeah.
So just you getting some extra, some spare.
Yep.
Did y'all slurp in there already? For the suction sample?
Nope. Just had time to get some water in there so far. We just measured temperatures in different places. So we checked the 2 other mesh boxes. So we did the work that needed to be done there. And then we came here, measured temperature, chose the warmest spot, and started sampling there an hour and 40 minutes ago.
What's the next thing we're doing?
The next thing here will be to do the same thing with the blue bag.
Okay.
Same corner. I was just very confused because I had a different view angle. It's because it's not the same camera.
Yeah, it's hard to see with that one.
So we've checked that thing. It hasn't moved much since we got here, but essentially we're aiming for right under the rock. There, in that corner. And that's where we found the highest temperature. Went up to 26 degrees at some point. Oops.
Okay.
A little too far. So that's where we will need to put the inlet for the blue bag
Got it.
in a few minutes, and that will be another 30. Also expel the water that is in there. So turn place it, expel the water, and suck back in that fluid there for 35 minutes. But Tihomir and someone else will be here while I eat.
Well, they let you eat?
I hope so. I get cranky when I'm hungry.
Hungry.
Sure. It's almost over for the water sampling. Did I mean to 76. Let me.
So you stop this now, like?
It's gonna stop in a minute.
Okay.
Literally. If I put down the time, it should stop at 17:56. See, it's out there. What time?
Yeah. Good.
So stop it, or we still got a little while?
I'll give it 30 more seconds.
30 more seconds, got it.
But it's looking pretty good volume wise. Yeah, we have to use the SciCam to turn around and see it, but it was looking very good.
Okay. Good.
Not overinflated and not flat.
Good. Then
Okay.
I guess we can stop the
Yep.
pump.
Okay, it's off.
Very good. And
return.
then we return this, and the blue bag sample will be exactly at this location.
Do you want to go have lunch or?
Yeah, I think I'll just log the sample, then I can.
Okay.
So Sabine, could I load this or?
It's the, oh,
It's
we
already-
just finished the pump.
No, the other one, the blue deck.
Oh.
We are sampling now. So it's 97.
Yeah, it's kind of melting my brain for Jack.
We're used to it. Alright, what's now? See, Uncle,
It might be too far. We lost it.
can we go back?
Yeah, come on back. Right
This will be the exact same location as before. Okay.
We should try vertical holsters for these sometimes too, you know?
We did that the first time.
And they sucked.
I don't know why we didn't like it. I think
I
part
know
of the
they
problem
stick
is
up.
it's so tall. Yeah.
They stick up right in the face, I know that.
Do it like your great grandpappy.
Okay, so I'll write this for some.
Cross route.
Yeah. Quite nice.
Nice. Alright, so now, you wanna do port arm? You want me to do port arm? Which one?
What ifs?
Just let me know if you want me to port out or anything.
They're just logging that sample out that they will do. From 97, and then from 98 on. Just press OK here. Submit. Yeah. Do you mind if I leave my computer here? Cheers.
What's that?
Before we start pulling on this, we might have to port back out and just make sure the hose is clear because it's around the side of that curve.
Tichy, did you put in water or not?
Yes,
Might be
just a little. He told me that just 30 seconds.
Never mind.
Just
We're
30 seconds.
There we go.
Okay. It's like a ninja that came out of nowhere.
So we first want to push out like the little water that's in the bag.
Okay. So first we're gonna expel.
Yes. But like only for like 30 seconds.
Can I tell something? Now we don't need turn off and on, just turn on and then output 30 seconds and back to input.
Okay, but then we do it directly inside because the time when we don't switch it off in between, then we also get water from the surrounding in there.
Okay,
So
they
we-
must switch
So first-
on, but
So we put it into the hole where it was with the other ones before, and then we expel, and then
Okay. You want me to take the starboard or how you wanna do it, Carl?
Here, I'll swap it. Yeah, the right side. Does that make sense?
it was very small distance, very little water. Like water.
If you, I don't know that I gave you a good grab for this angle. The arm's gonna be blocking your view. So I don't know if you want another, like I can help you get a better grab if you would like before. And then the other reason is because that pushes you to the right, and now you're blocking my access for the valve, for the switch. Awesome.
How about I give it the old college dropout try?
Good.
And then we'll be What do you think about it? You know?
Just remember that that needs to be the very top of the wall, the cavity. So with the down pitch, I'm worried that'll be not as
I'll
good.
tilt it up more. I just don't want to too close and whack it, you know? Can you zoom a little on that?
I'm gonna do that.
That's it. Yeah. Something like that.
Yeah, just, to me it looks like it's pitched way down, so I don't think your tip is at the roof, which is kind of important. It's getting better.
They're kind of important.
I know these things sometimes do. Okay, that's a lot better.
Everybody's liking it?
And I think I can get over your arm now for the, yeah, definitely over your arm. That'll work.
Froze?
Is this gonna be an example?
Yes.
Something like this,
The knock it?
This is the exact place we've been before, because I was not in before. That's good.
It's a little deeper in
Yeah,
there,
but like the same
yes.
location. Very good.
So yes, but no.
Yes, but
It's yes, yes, and yeah.
okay. Oh, we covered the writing. So it's in back position right now, right?
Yes, now to auto back. Right.
Is the camera all the way?
I can get a lot of it. Yeah.
Or at least like half of it.
All the way.
Okay. It's work. It's
That thing's blocking.
It's work. It's work.
That's good?
Yeah.
Okay.
Oh, 30 seconds.
Probably pretty close to 30 seconds by the time I go and grab that other one.
Okay.
Good?
Yes.
Okay.
Give you an idea of the close quarters. Routes will work there. Got a picture.
Leave it.
Yeah, I'm not gonna do it.
And now turn on the back.
So using the arm to reach back inside the sub and hit those controls for the pump.
Okay. If you want now it's little bit on, this little bit left. Not to,
Turn more on.
Yes.
Okay.
But it's not work. It start very fast. Okay. Yes.
And you can see our science cam in the bottom right hand corner,
35 minutes.
and the view it's showing in the picture-in-picture.
Okay.
Sabina, this is sample 98.
This is sample number 97.
Okay, so the 105.
that current is a lot smaller than I thought it would be. I think I would have though. There's so many of these things.
I'm sorry, I have to put you in, you got it.
Maybe this work.
I think it's good.
All right, if you're just joining us, these are live images from the bottom of the Pacific Ocean. You're looking at a live feed from ROV Sebastian, Schmidt Ocean Institute's remotely operated vehicle that's deployed from a research vessel Falcor 2. This is the Underworld of Hydrothermal Vents expedition, and we are on the East Pacific Rise, about 1,000 kilometers south of Mexico and about 2,000 kilometers to the west of Costa Rica. So the team has been deploying various experiments, and we're in the process of recovering some of those experiments, but today we're doing some sampling. I did, looks like we're filling up a pump here and giving you some views of the seafloor. And we're back in the same spot we were yesterday, right guys? We're, you know. Go ahead, Carl.
No, I just said, yes, this
Oh,
is it.
I thought you were going to give us a detailed explanation of what's going on here.
Wrong person.
Wrong man for the job. Carl,
He could, he's just not hard.
I know, he's not feeling very chatty today. So yesterday, the team pried up a little bit of the seafloor, which had been a site of an ongoing experiment during this expedition. And this expedition is called the Underworld of Hydrothermal Vents. And as the principal investigator put it, we then were looking at the underworld of hydrothermal vents. And so they pried up a little bit of the seafloor, and we had a great view of what's going on underneath. And it was a venting site, and we've returned to that site to take some more samples.
That's there.
That's it. That is the underworld.
So styrofoam.
And if you missed the excitement yesterday, the science party was thrilled because the underside of the slab they pried up did confirm their questions they were trying to answer on this expedition, that the Riftia will travel through the hydrothermal vents to recolonize areas.
Can I take the camera? Ah, that's it, okay. This is in the cave.
Is it one of these?
No. So this is a tube, but this is not belonging to. So this is not from this polyheat. It's very difficult to see.
Let me see if I can get you a view in the T4K.
It's difficult to see now, like what has fallen in. It's like one of the.
That is. I don't know if I'll be able to get close enough. Maybe.
So Chill's going to take us on a tour here. The camera on the end of the manipulator.
Do you want me to zoom out so you can kind of see where it is? Well, I see your manip.
Am I in the right?
This camera obviously is not 4K.
Yes.
I think I'm too high, am I not?
But I think the problem is that you would have to come where my arm is and like look that way. But it's like, it's right below where your jaw is, you know? But it's gonna be really hard to see it. It's,
Yeah, I don't know that I can see it.
I think if I didn't have my big stiffener on right in the way.
Yeah. They're
Like usual.
Always in the way.
Like usual. Just messing y'all up, man.
Yeah. I'm in the area, but I just can't see them.
Oh, you're not.
Okay. What else can we explore?
You know what's funny is yesterday you did like that rock. You basically did a table flip emoji.
Yeah. That was really great. Wow.
Yeah, that was actually fun.
Does that camera focus pretty close? Like, can you get like right on one of those worms on the rock? Like,
Sure, I can.
The kind of one with these are just, you know, the pieces that were falling down. So I think this is like just from the surface.
Yeah.
And it's also black, so it's like a,
yeah, for sure.
yeah.
I don't know that I can get closer to the 4K zoom.
Yeah, it's got quite a good zoom. You're pretty close though.
It's touching there.
That's with the lights on? Or no?
I don't think I have the lights.
Not too bad. Imagine how cool it would be if we got a like a 4K on that.
Yeah,
That would be cool, yeah.
that's lights calling out. Yeah, no,
But probably it could be one of the Serpolites. Could be. Yeah, I think it's just the rock that tumbled down.
I can give a selfie.
Yeah, 4K on that camera would be amazing.
There's Carl's looking into the sun. The lights on Sebastian. There it is, the ROV selfie.
Unfortunately, it's upside down. That's the best I can do.
Do you think those are good ones?
Sorry?
Do you think those are young tube ones?
No, so I think like it's just like here that yeah, the rock pieces, yeah, just falling, and I think it's just probably it's one of the Serpolites we see. There's also another tube one probably on, but I think this is just from the from the surface. So,
Maybe a 4K with HD, that would be, you got a lot of cool stuff with that.
I don't know, you just run the cable externally, you mean?
Well, you can send HD over SDI, digital signal over SDI, which is just coax.
So then you're talking about using that housing to put a 4K?
Or HD.
I wonder if you could get another housing with like a bad camera. Those are a dime a dozen, can't you?
Yeah.
You know?
It's just a welding camera. We have two of the spare welding cameras in there. As long as it matches those dimensions, you can just put a webcam, tiny webcam or something. SDI for everything.
That'd be so cool.
That'd be cool. So Sabina, how was your night last night? Getting that rock in there, and what was that like?
so you know, the entire afternoon was so exciting that it was like completely like dead in the evening. It was like I don't know, there was so much excitement that I don't know. I was like really tired yesterday. Later, but then we worked through samples, and it was really cool to get the rock up. But it's like, you know, when you have all this tension and then you find this and like you, I don't know, at least then I like afterwards I collapse.
Yeah. So you guys feel
But
more
I had a good night, so now I'm like
ready
back.
for more, huh? Do you feel more relaxed now that you found that? Or are you guys still back to business as usual?
I guess like both. So you kind of you also sometimes have to just switch your brain to like a working mode. So you have like all this excitement, all these findings, but then you kind of also have to force yourself to switch back on working mode because otherwise you would mess up the next days and the program that you're currently in.
Yeah.
So you kind of at the same time you're still the excitement and everything, but at the other time you have to continue. Otherwise you cannot finish the whole picture and the entire story.
Gotcha.
But like yesterday was like the day basically.
Yeah.
And now we have to continue to kind of make everything like a proof and so still a couple of very exciting days ahead. What was a pity yesterday that we lost the blue bag with the water from the mesh box. So that's really not that is that was a pity, but we will have like 2 more chances.
Okay.
Which is good. So that's also why we make 3 experiments to do this. And yesterday was just like really also a lot of also luck. We had a lot of luck. Of course, there was a reason behind why we selected this site. A lot of thinking behind. But like this finding yesterday, just it blew my mind. It's like just so cool. And then also the working together here with it's just cracking and flipping over. It's fantastic. It's really really cool.
So there's a lot of excitement in the labs last night till late,
yes,
right?
Yeah.
So I was curious about something last night. Maybe I'm just underthinking it, but when we first flipped these rocks out of here and exposed this cavity, that area looked to be covered in what looked to me like the same bacteria that was growing inside the box. I could be wrong about that, but that's what it looked like. So my thought was, it was, before we uncovered this, was that cavity full of that bacteria already? And then it came into the box that direction? Or by us putting the box, did we create the conditions for the bacteria to grow and it went into that cavity from the box?
So basically what you have like in this subsea fluent that's known for a longer time that there's bacteria that can live in these systems and they always come out, but usually you don't catch them. You don't visually see it, and by putting this mesh box over it, they cannot, because they're also rather large, these filamentous bacteria, like they form these clumps, so they are getting caught basically. So they are still produced like down there, but they cannot escape into the ocean.
Sure. So you think they were in that cavity before?
Yes, but there's like constantly vent flow coming, so there's constantly energy for them to produce. So they produce all the time. But normally they would go into the ocean water, and you wouldn't recognize it in this abundance. And this time you kind of, it's also very interesting that Stefan Sievert, like he's working on microbial communities. He actually told that they once like tried to design something to catch that these bacteria, microbial communities from down there, but he thinks that actually this design is like much better to kind of capture this.
Sure. Yeah.
This is the same bacteria that becomes symbiotic with the Riftia, or is this a different one?
That's a different bacteria, so it's like different, bacterial species. And so for the Riftia, it's like it's called it has a special name because it's only found in this tubes and the Riftia.
These bacteria under here are just purely chemosynthetic.
Yes.
So they're kind of bottom of the food chain, basically.
That's also the reason why we find all this abundant life, because all the bacteria can use the energy from the vent fluids or the sulfide, but also other chemicals. And basically, what the plants are doing on land using the energy of the sunlight, these bacteria do this with the chemical energy from the vent fluids and produce biomass.
I was thinking after we popped that rock, seeing how much of that there was. I mean, there
It's
must
a
be
lot.
some significant
Yeah.
biomass under
Yes.
this, huh?
Yeah.
Have we already done a lot of studying on that particular bacteria, or is this something that's still that we're learning with the collection that we got yesterday?
I think also because I see Stefan Sievert is very excited, so I think we are still like learning a lot. But but this kind of, it's also called like this snow blower events, so like documented since already, I don't know like when, since a long time they're known, a long time, it's relative, but sometimes you see this massive like white bacteria like flocculants basically in the water column here, and it also means probably sometimes they can erupt.
Oh, we saw that
And,
earlier.
yeah.
That's probably what we saw in the look back.
So, but like I don't know, like how much biomass there is like in this underworld. I think we don't know. But looking at this, it must be a lot.
I wonder if there's any
Yeah.
critters that live under there that we haven't seen
Yeah,
yet.
this is what we want to find out by the blue bag sample.
Oh.
And also by the sample that we're taking today, like this one and also tomorrow. And yeah.
So can you guys do like a DNA analysis
Mhm.
on this water and say, oh, that's new?
Yes, also it's also the reason why we take like all these control samples. So we had, we take samples like among the tube bombs in the water, and then we also have these samples from the subsurface, and then we can compare basically the diversity and can identify like potentially species that can only live in the subsea floor. So that's the reason why we do all these different measurements and collections.
So if you're following along yesterday, when they pried up that slab of seafloor, a lot of bacteria came pouring out. And they'd also trapped that on the mesh box that was set
Do you
down.
know how much bacteria?
And so
there?
Dr. Schmidt
Like
Ocean
in
is
the
saying that
Subsea floor?
Subsea floor? Are there any estimates in that?
Yeah, we have
Oh.
an estimate actually in the
Hold on.
paper that we published a few years ago. And so, we looked at the activity
Mhm.
of the microbes. And the productivity is similar in the subsea floor as it is on the surface. So half of the production appears to be happening below the seafloor.
Okay, oh,
vents.
that's a lot. Yeah.
And then we also estimated the amount of standing stock of biomass
Mhm.
that's actually below the seafloor potentially to support that
Yeah.
productivity. I don't have the number on top of my head right now, but
But about half of this is like in the
productivity-wise, yeah,
mhm.
well, approximately, yeah.
And do you know if this is like special for this for the 9 North EPR, or you would also like expect like similar things in
Yeah.
other
Yeah.
regions, also like in lower spreading centers?
Yeah, I think we would expect similar.
Yeah.
Yeah, I think depending on the subsea floor conditions, of course,
Mm,
but
yeah.
in general. And in some other places it might be, here it's mostly sulfide oxidation,
Mhm.
and in other places it might be hydrogen, depending on the geochemistry. But basically, I think what we observed yesterday when the box was removed, like the snow out,
Yeah.
that's very similar to what they observed after the 91 eruption.
Yeah.
There was like a lot of snowblower activity coming from the seafloor.
There's some that right there that probably blew out. That's that stuff, huh?
So we're thinking that's some of that bacteria.
She's shaking her head, yes.
Yeah, these are like flock. I mean, you mean what? The flock here. I mean, a lot of it is also sulfur. So these organisms oxidize hydrogen sulfide with oxygen, and then they form elemental sulfur as an oxidation product.
And the type of shrimps that is swimming in the flow, these are amphipods or what?
The small critters? Yeah,
Yeah, these are small.
amphipods.
Yeah.
So it's very popular.
They really track the flow of the fluids coming out.
Popped up an image of the bag that Sabina was describing earlier. Using that to take samples of the seawater and vent fluid, and taking samples and looking for maybe new organisms that are unknown from beneath hydrothermal vents. So we're slowly filling that bag. Got a little bit of time. And we're swapping in a new pilot.
Just be wary if you did start to move to watch this wand, you know? Wand and arm coming down in that hole. She's been stable up.
So now we got Alex on the sticks. Jill is co-pilot. And he's doing a stellar job right now. Dr. Spienegal in there in the science chair, and this is Tyler, one of our marine techs for this expedition, talking to you right now.
Different bag, some action. Different bag, some action.
Yes.
How much more time do we have on this?
10.
And Salve in the chat bringing up the origin of that MBSG. So earlier the team was discussing the chemosynthetic bacteria, and when they pried up that slab there was a bit of a snowstorm of bacteria coming out. And if you caught the discussion earlier, that is a different type of bacteria than what is symbiotically connected to the Riftia tube worms that we see in our picture here, those white tubes with the red plumes. So those tube worms don't have a mouth or a stomach. They rely on the chemosynthetic bacteria that is symbiont with them for their energy. So down here, organisms can live without sunlight. So those amphipods, Sabina, the little critters in the shimmer there, are those amphipods? Is that what they're called? Okay.
Are they symbiotic?
No, not that we know of. So they are feeding, on what exactly we also don't know. Likely also on the bacterial mats, or the bacteria that are also in the water potentially. But they're very abundant here, so wherever, we've seen them a lot, and there are currently like thousands and thousands and thousands. Millions, I guess, on this vent pit.
Yeah, there's swarms of them in places. A constant reminder of the density of life down at these hydrothermal vents. And just outside of these, it's been described as a kind of a desert on the seafloor. So in frame we have a lot of different types of life, and there's a lot going on down here. And that life is fed by the hydrothermal vents, and you can see the shimmer on the screen here, and that is vent fluid coming up.
So this aphicos has host no symbiosis.
No, not that I know of.
So the scientists saying that they don't believe the amphipods host symbiotic bacteria. And these are images from 2.5 kilometers below the Pacific Ocean. And here's a view of the surface. So this is out the hangar door on Falcor 2. Beautiful day on the East Pacific Rise. We've been seeing oceanic white-tip sharks. Yesterday there was a sizable manta ray circling the ship for a while. So various crew members have some pretty good video of that. And I think it appeared in the end of the dive, right? Did you guys catch it on the RV?
We got a lot of video.
So if you stay tuned till the end yesterday, you saw that manta ray that's been circling Falcor 2. Is it coming right for us?
We did, yeah. We had a kind of a perfect scenario. We had the current coming up from behind the vehicle as we were backing up for recovery. And so that created a situation where he was able to feel like he was swimming towards us, even though we were both kind of stationary. And so that made it where he was just steady flying in front of the RV in the perfect spot in front of the camera. It was actually pretty epic.
Sounds epic. If you were in the control room, Alex and I were on the winch, so we saw none of that.
Dang it. Got some video for you.
We'll have to review the footage. So on the previous expeditions, Joe, were you guys seeing as much large like sharks and rays in the water around the ship?
No. We saw some mahi was about probably the biggest stuff that we were seeing near the surface.
Yeah, and you saw mahi up close once?
Yeah.
Briefly?
Yeah. Oh yeah. Yeah, I did.
Yeah, saw a lot of mahi the last expedition.
Sure did.
We did.
Sure did. Some more than others.
Yeah. Some famous mahi. Yeah, it's pretty cool.
Yeah.
To be out this far and see those creatures swimming around. Did you see the manta, Sabina?
No, I was in this room here and excited about other things. But I've seen it also like in the video, like when the rough was up. It was really nice.
It's cool.
They have pilotfish also.
Yeah, swimming around right underneath it, right?
Yeah, more symbiosis.
Yeah, and we saw last expedition dolphins with what are the ones that attach?
Remora.
Yeah, remoras on them.
And the other day there was also a hammerhead shark on the side of the ship.
Who saw a hammerhead?
Jono did. He was talking
Really?
about it, right? Did you see it too then?
I didn't. I love them. I wanted to sit and I just arrived too late.
Wow.
Just have to stop
That's the way
sleeping.
it is.
Just walk around the ship all the time. Yeah, so oceanic white-tip, hammerhead sharks, manta rays.
And one flying fish that came on board.
Yeah. We get those from time to time.
It looks like what we've been calling a manta ray may not have been a manta. But I'm not an expert by any stretch of the imagination. And we'll bring it back down to the seafloor. So again, right now we are filling this bag. It looks like it's filling all right. I don't see a doodle on this one though. We added some artwork on previous bags.
Sabine, do you think the community of biofauna in the crust will be very different?
I have no idea. That's why I would love to look into what is now in this blue bag. So we'll hopefully find out later today.
So come out. I think it might be, I'm not sure.
Camera feels safe there.
It's all the way in. Should be.
Scientists excited to examine the contents of this blue bag when it gets up to the surface.
Alright with that light.
Yeah, it's fine.
So the RV pilots are gonna shut it down. Whenever the timer goes off. Gonna shut it down. So they are pumping for a specified amount of time. And once we hit that time, the team will finish the flow into the bag, get that buttoned up as best as they can, and we'll continue on with our dive. You got the manipulator view of the what's going on there.
With that, the bottle is out.
And then I'll give this a little push again, just to make sure.
Right on top of the other armor. So we should do too.
Is that the way this is supposed to be?
Oh shit. Nice.
Oh, that's right. Wow. seems to work.
So what do you? Let's see what we got.
Light.
Yeah, actually that would be better if you can reach that with that front instead.
I don't know if Russ told you, but he's put a new blade on the.
He did, and I forgot about it, and it works amazing.
Yeah, it definitely cuts. Sorry about that, dude.
It's all good. He told me. Yeah, let's
Anything with that hook
try
on?
that.
I don't know that I'm going to be able to hope so with the science game, so I can't get over here.
Well, how the hooks? Eventually, we're gonna have to take the bungee off.
You can put this also in one of the bio boxes if you want to.
It'll float up.
Well, even the lower hook.
Oh, the lower hook. Yeah, let's try that. Dang it.
There's a loose spot right there. It's quite confusing to zoom on that.
Jill's trying to skewer that monkey's fist on a hook.
Oh, I'm going the wrong way. It's like, oh, look. That's why.
It's like threading a needle from 2.5 kilometers away.
I think you got it.
Good job, Joe.
There it is. So is that bungee to secure the pump in the off position? All right. So a bit of a new addition. Team installed a bungee to hopefully hold that handle in the off position on the pump, so we don't lose any of the sampled water and vent fluid. And on the right manip, we typically have coral cutters. So it's like a knife edge so that you can shear off objects to sample. And with that right manip, if you're using to pick up rocks or other
Looks like I'm hitting the levers.
hard stuff, it can quickly wear the edge off that coral cutter or break it. So the team put a fresh one on there and it is
On that box
sharp.
too much. On it a little bit at least.
Oh, you're trying to grab the tube. Lower that.
Is there a tube?
Yeah, it was inside of the braided tube, like the same hose that we had in the section. But
I thought the tube part didn't move.
it didn't. You can see the yellow, like what's left of the rope up above.
Yeah. This is awesome.
You want a little bit of this again?
Oh.
Nice.
See, the tube is fixed.
Yeah.
So
The
I
bungee's-
cannot grab that. Come on.
So Sebastian is a sizable vehicle, but there's only so much working space the team can use to implement the tools and devices and various controls.
Well, I see what we're supposed to do.
And that was essentially a blind grab, so that's a pretty good grip that Jill pulled off there. But in configuring the porch and the sub for every dive, you have to find a place to put different things, and so the spot this bungee was placed was not in the most visible spots, but the team's doing what they can do, and you can only put things so many places.
So stuck right now.
I didn't think you were on the camera.
Can't pitch it up anymore to get over that. Yeah, I'm just literally maxed out. It won't go up.
It's like stuck somewhere below.
That's gonna do nothing.
No, what about the back? I don't know that move, but.
So we triggered this Q93.
Mission accomplished.
So it's set 198.
She would have did 1,000 meter depth.
Yes, but this is.
Yeah, I know.
It's not a problem.
I'm gonna have to see with her if she wants the two other ones here. She will shoot it. Okay, thanks.
So we mentioned before, nothing is easy in the deep sea. And these diodes are a constant work in progress and problem solving.
Good. So we can put the
Take the probe?
yeah.
Okay. Okay, can we push out?
Come on.
Okay. There's the entrance.
Was there upholstery?
dice, I would like the two other ones triggered here as well, so that she can have full sample. And she'll do the 1,000 meters.
You want to?
It's up to you. Seem to be having fun.
It's nice to see you.
This is yours.
So,
We need suction sample here. Fine, super fine.
So as we were just discussing on kitting out the sub for these dives, there's limited space to put a lot of equipment. So we got our bio boxes, we got that cooler up there now, got a couple of pumps, heat temp probe, suction sampler, coral quivers. I don't think we have any push cores out there now, but there's limited real estate to put things and places to safely stow things. So another challenge of doing these deep sea scientific dives with an ROV.
We'll want to trigger the two other Niskins here so that she has a full set here as well.
Which one fired, do you know?
The three fired, right?
Yes, 3.
Okay. I saw dab on your face and I was like,
Team's gonna fire off a couple Niskin bottles here. We've got these triggers set up so the pilots can hit them with manipulators.
yeah.
So we've got the very helpful visual of when the flag pops. The Niskin has triggered.
You good with the numbers?
99, sample 99, sample 100.
Great.
So that's bottles 2 and 1. Those Niskins are like tubes on the side of Sebastian you can't see. But they will trap seawater in there.
Sample 99.
So we bring them up and the scientists can use that as another
98
water
is
sample
Niskin.
in their experiments. And that was your design with the flags, right, Chill? The Niskin's triggers?
The flags were the original levers were, I think, Russ, slightly modified, but yeah, the flags were mine.
So a lot of times what you see on the sub has also been designed and installed by the pilots, and that is one of those systems that they put together to make the collection of Niskin samples easier and more efficient. Pretty cool.
So we're gonna do some suction sampling here now, guys. And we start with this super fine.
4, 5.
So
5.
4 or 5.
5 looks like it's already been.
6 was used. 5 wasn't.
Yeah.
Shouldn't.
Yep.
Is that right?
I noticed from the X.
Okay. Oh, the exit was just Monica was
Okay.
counting on this.
Got it. So you want to go to.
So 4 or 5, whichever is more
It's on
convenient.
five.
It's on 5. So we're
Oh,
good.
let's stay on 5 then. It's good.
So we got a
Very good.
new scientist on the science mic. Stefan, introduce yourself
What were we gonna do?
so they can put a name to the voice.
Hello everyone, my name is Stéphane Hordez. I am a researcher in France at CNRS. I'm a deep sea biologist and I study the evolution of adaptation to extreme environments. And on this cruise, I'm one of the participants. I was involved in measuring the chemistry of the seawater, but now I'm just doing something else because the mass spec had some trouble during the first dive and got flooded. So trying to fix it still, but for the time being it's not working, so we are sampling water in little bags for that.
And you've been working feverishly. See you often hunched over that mass spec working on it.
Yeah, it's tedious. I'm at the point now where you have to let it work on its own for a while to try to dry it up inside, and that takes time. So it's happening, and I need to keep an eye on it all the time. So here we're gonna use the suction sampler to try to characterize the fauna we have in this hole we opened yesterday. So we'll be suctioning a little bit with 3 different mesh sizes. So right now we are in a super fine mesh, and we're gonna go down at the bottom of this hole and just suck some water. As you can see, it's coming up. So we're gonna look at the animals that are found in there, also at the microbiome that is found in there.
Are you wanting that bacteria or?
Yeah. Stefan Sievert was very interested in this bacterial mat as well, so that's what we're looking at right now, coming up very easily off the pebble bed there. And we're gonna try to see whether we can go a little bit under the overhangs as well, but it's a tight spot, so we'll see if we can get there. Nothing's guaranteed, of course. In the meantime, we're seeing the this fluff coming into the jar in which we are collecting, so we're pretty sure it's making its way up there. The ultrafine mesh is about 32 micrometers, so it gets clogged pretty easily, so we will lose power, suction power there a little bit. As it happens, then we'll just switch to another jar with a slightly bigger mesh and a third one with even bigger one.
So we got a shot of the jar up on the screen, so you can watch that bacteria get trapped in the suction jar.
We still have some suction there. You can see the mat coming up, hopefully with small organisms as well, to look at the myofauna. But
so typically when using the suction sampler, the co-pilot will aim the nozzle where it needs to go, and the pilot will work on the pump and switch the jars and run the camera.
so it looks like we have very little power, suction power left. So we're gonna wait 1 or 2 minutes in the water to clear the tubing, and then we'll switch to a larger mesh jar. Number 101. And that will be jars.
Suction, and then get ready to just turn it off. As soon as it starts rotating, then turn it off. Try not to go to 6.
I've already got a sample.
Exactly.
I don't remember which blades,
I don't. Yeah, but if it goes to 6, it's okay because you'll have a little bit of suction, so you just hold everything in there and you just keep rotating the next. Just don't stay on it. Yeah, just rotate past it.
And is it working now?
It is, yeah.
Oh, without the top.
So just rotate it to low pressure, and then you're ready to just push it on.
Yeah, right when it starts.
Yep.
If you wanted to run that to about
So we're working to clear the line here.
So we're gonna go to either 1, 2, or 3, whichever is more convenient for you.
It'll depend which way it starts spinning.
Okay.
You ready to, you're gonna let it keep going a little bit?
Yeah, maybe a little bit longer. There's still particles coming. And there, sometimes big particles. It takes a little while because the suction power is relatively low there. But whatever stays in tubes will make it into our next size sample, so it's no huge deal.
Okay. No. The ocean will fix.
So can
this bacteria survive in lower temperatures away from the vents?
Yeah, survival absolutely, but it will eventually require to have some of its food, essentially sulfide and oxygen at the same time.
So once it gets far from the vents, it just becomes food for something else.
Exactly, exactly. It's just a major organic matter export from the vent to the surrounding area.
So for filter feeders and fish.
Absolutely. Absolutely. Filter feeders, deposit feeders as well. So the sea cucumbers, when you see them landing with their pops, they're gonna just kind of vacuum the surface of the rocks and just get their food from that.
Yeah. That looks like it's starting to, well, let's say that more so.
So I think we're good. We can switch jar, please.
Okay, just real slow. Indexing to 1, 2, 3.
Just skip that one. It's 4, 3. Perfect. Thank you. So we now switch to jar number 3. I think the mesh size in there it's 100 microns. So for the mass spec, it was indeed a bad surprise. Essentially, the membrane that should have been watertight was not, and water just got into the vacuum chamber. So it's why it's such a pain to try to fix it. Got into all the pumps. So that will be 102 in jar 3. No, this one. That's okay. If you can brush it inside, but we don't see anything, so I know it's not obvious. But maybe if you can just move it a little bit inside, under the sorry, I mean that's it. Yeah. So we dislodge some stuff. I see a scale worm there, the red one there. Yes, I know. Typical, Stefan. Typical. If you can get it. It's interesting to know that these guys are down there.
Aim for a little red scale worm.
I didn't understand that.
Okay. Okay.
It's still there. It may be in there. So, no bag of rice. It would have been funny and an easy way out. The water got inside the vacuum chamber, but not inside the housing. There was very little seawater inside the housing, which is a lifesaver there because none of the electronics actually got exposed. Only the inside of the vacuum pump. So we'll figure it out. Probably not before the end of the cruise, though. No guarantees. Someone on the chat is asking me questions. I'm not talking about Connor. Tell them I'm not talking about to someone in my head.
That's cool. You see the broken pieces of the where it was dripping down the lava.
Yeah.
They were like loose.
The little stalactites fell off there, right there. It's pretty good suction there. It's good. Nice sample. Look at that coming in. There's a snowstorm in the jar right now.
Look what I found.
Nice.
Oh, numbers.
So that looked like, to me, like it was another small layer and like a cavity of bacteria just underneath it as well.
Yeah. So Stefan Sievert earlier was saying that it was strange because a lot of what he had seen yesterday didn't look like it was still there. So it could have floated away slowly, but probably that big pile was part of it as well.
Yeah.
Still some good suction here. Not clogging as fast as the previous jar. And we still need to do one more jar with the coarser mesh size.
I can flip another rock out here pretty easy.
Let's not do that just yet. I don't know if we have enough in that jar. I'm pretty sure we do. So we'll just hold it in the water for a little while to let it clear. So, later. Pretty clear here, so we're gonna switch jars and we want to aim for jar 7.
And while the pilots are getting the section sampler ready to go, quick recap. So we are
Good. Thank you.
on the East Pacific Rise. These are live images from ROV Sebastian. And this is the Underworld of Hydrothermal
I
Events
think you can reach there.
expedition.
Yeah, let me see what I can do.
Yep. Give it a shot, please. Otherwise, probably here would be okay as well.
Good, just go for some science.
We got Dr. Stefan Ordez in the science chair. Alex is our pilot. Chill is the co-pilot. Use the manipulator to get that suction sampler right in the exposed hydrothermal vent that we pried up yesterday. And I am Tyler, one of the marine techs on this expedition, talking to you right now.
Yeah, not an easy reach there.
Yeah, I mean, it's getting some of it, but it's a smaller gap on that side.
Yeah. Can you try on the left-hand side where we're measuring the temperature? Maybe? There.
So this is the site of Experiment 1, which is a dye box experiment the team deployed a few days ago, and yesterday they retrieved that mesh box and in some sampling pried up a chunk of the seafloor, which revealed evidence that tube worms were living beneath the hydrothermal vent, beneath the rock they pried up, confirming their suspicion that these organisms can travel through the hydrothermal vents to move from place to place. So we've returned to that site here today to do some more sampling. And previously they did a bag sample, pumping some seawater and vent fluid into a 55-liter bag. Now we're doing some suction sampling to see what we can pick up here.
Ooh. Look up. We got that thing way up over there. Look up a little bit. Yeah. You can tell on the sit. Oh, maybe I'm not as far. No, I'm in there.
Yeah, good. I'll see what comes out.
So with the view of the sit cam, our main screen here, you can see that Chill's got the nozzle of that suction sampler wedged as far underneath that rock as he possibly can.
Yeah, maybe in the rubble on the right-hand side as well. A little bit there, yeah. That could work at the base there. Go straight down, maybe. Nice.
Look at the jar.
HD4 too.
Yeah. It's just a giant cloud of bacteria. Sebastian's gonna be sicky boy when he gets on there.
Agreed.
Get my rinse down.
It's either that or it'll be immune to everything.
It'll be invulnerable.
So bacteria smoke.
Yes, coarse mesh, yes.
So if you're watching yesterday when they pried that stone up, it was like a blizzard of bacteria, and in the mesh box as well. So the team is doing a good job suction sampling as much of that as they can, and anything else that gets caught in the nozzle.
Rubble. Down. That's okay. We don't want them at this time. I think we're pretty good. Hold it up in the water for a little while for the pebbles to fall off and the water to clear.
So Sebastian is slightly positively buoyant. So without thrusters it would make its way to the surface. So to set down on the seafloor, the pilots keep some constant thrust down to keep it pinned to the seafloor, so to speak. So when the manipulator pushes against the ground there, oftentimes you see the sub rock back and forth. It's a bit of a balancing act to get the right amount of down thrust without too much, so we don't raise the temperatures too high.
Okay, good. I think we can turn that off. Or turn.
So I'm just gonna turn it to 18.
Yeah, to 8 so that it's.
So when they're done with a suction sample, they'll swap it to a different jar, and that effectively will seal the jar they're sampling into.
Okay. So here we still need to pick up some rocks for Avery that we will place into BioBox 4.
Pilots are working to stow the suction sampler because it's got that long tube. We don't want it dragging along behind us or in front of us.
So we're gonna get rocks, you said that?
Yes. No, from the bottom.
When we get closer, so you get the port on.
Yeah.
You can probably want to use a scoop or something.
No, we'll try the port first.
Okay, okay.
Or you, this might be good if you want a bunch of like small loose stuff.
Yeah. Could Fanny, could you find Avery and ask her whether she wants pebbles or rock?
Okay.
Thank you. While we do that. On the to-do list, there's also to place the frame next to mesh box 2, but that is already the case, right? So probably don't need to do that unless.
So while the scientists confer about the size of the rocks they'd like to sample. The pilots are setting this up a little bit closer to make that collection easier.
In the meantime, I'm gonna have a quick look at the place we cleaned to see if there are any crawlers down there after that.
Stefan's going to check out the freshly vacuumed hole.
Pebbles.
Pebbles. So I guess scooped in, right? Sorry, I'm gonna zoom out so they can see what you do.
Pebbles is the call, so the team's going to grab this scoop, make that the easiest method for sampling this. For larger things, they probably just use the manipulator itself. So they're going to bring the porch out
Thank you, Fanny.
so they can grab that scoop.
There's really nothing to scoop against. Let's see what happens.
Stefan, where do the sample go?
You will go into Biobox 4.
Oh, you're trying to get, yeah, cool.
Pebbles.
Yes,
that's
I think
what you
that's
want.
it. Yeah, that's great. That would be perfect. Yes, nice. But it's different from yesterday. There was a lot of like yellow stuff.
Yesterday or
Bottom?
today?
Yesterday, when we
Okay.
when we left.
When we
Seems
used
like
the
the
suction
section-
sampler, there was a thick layer of that
Oh,
at the
oh,
bottom. So that may be
oh.
it.
So you were able to slurp that?
Oh yeah.
Okay. I need
Ultra
to
fine,
go back.
fine, and coarse.
Ooh, I need to go back and.
I think that's all you want, right, Avery?
Yeah.
You don't need
that's perfect. Yeah.
Okay.
Yes,
So
pretty good.
Yeah.
This
So was
subsurface
Monica
rock.
trying to get some of the background basalts today, or was she doing the?
I think that might happen later, right? Still.
Oh, gotcha. Okay.
I don't have that on the list,
But
but
I think it was mentioned yesterday in our discussions that she wanted to maybe get a bare basalt rock already today.
right.
I wasn't sure if that was today or tomorrow that she was saying that for.
For the time being, these in Biobox 4 is good, right?
Yes, that's great.
Okay.
Thank you.
And it's pretty watertight, so it should be good.
Perfect.
Oh, okay. There won't be anything else going into that.
That's it.
Okay,
Oh, okay.
It was a clean.
nice.
We just throw a refrigerator in
So
there.
it's like subsurface
Yeah.
rocks from explosives as well.
So obviously not all the scientists can be in mission control all the time. So often we'll have scientists ducking in and out, requesting samples, overseeing the collection of some specific samples they'd like, and then they head right back to the lab.
Stowing the scoop. Thank you. Is Monica around? No. Okay, not over either, right? Maybe computer room?
Should I go for it?
It's just we have time to go to Bio 9, so I was thinking about going to Bio 9. Say that again?
Could you take some work with the water or no?
No, piece of tuning
Fish up chin.
into a quiver.
Should I go for Monica?
130. Oh, it's all good.
So while the
We'll
team
go there.
works to get everything stowed there, take a quick look in our aft camera, and you can see it's a different landscape already than what we're looking at in front of the sub. So the density of life drops off really quick once we get away from these vents.
So let's pick up that frame anyway and put it there. It's not very far, it's just right next to it. And just get it closer to mesh box 2.
Sure.
Please. The bag is not moving. It's good.
Yeah, yeah.
Is 2 the one that's closer to us?
2 is the straight ahead of us. Just put it down there, maybe on the right hand side. Oh, not where you guys will need to work tomorrow. That's the one thing we have to take into account here.
Well, we're gonna have to figure out exactly where we want to sit down tomorrow. Anyways.
Okay.
So I'm just gonna come sit down. We're not gonna squash too many critters.
Okay. Sounds good.
So we got some tight quarters down here at the experiment sites. So we have 3 of our own experiments down here. You can see evidence of prior submersible visits.
Yeah.
And the team's trying to find a good place to set that frame down and also keep an eye on the organisms on the seafloor,
Could have been done
trying to
tomorrow
disturb them
anyway,
too
man.
much while they set down.
That's good. Excellent. Thank you. So we'll now head to Bio 9. which is south of here. It's out of the main Tika field, so it's gonna be halfway there.
Oh, we're going all the way down
Yeah, it's probably gonna be about 20-minute transit, I think, something around that.
here. So we gotta get that's
Sorry, where are we going?
all the way to the bottom one.
Bio 9.
Oh, perfect, okay.
So a little bit of travel there. We'll see black smoker again soon. And there what we will want to do is measure temperature, take a piece of chimney and put it into a quiver and come back here after that.
So we're going to do a little bit of a transit here. So up on the screen, got a map that the co-pilot's looking at. That Bio 9 is the yellow marker at the bottom of the screen.
Can we make it a little bit of a track here?
Yeah, that's all copied.
And you can hear the pilots calling in the
Yeah,
waypoint
we
move
can
for the
get
ship.
CD after.
1, 2. How's it 0.2 south?
Perfect.
So the pilots are coordinating with the bridge on moving Falcor 2,
Straight there.
getting the
Absolutely.
speed of the ship dialed in because Sebastian wants to be a specific distance out, safe distance. So they're working together.
Fast, just to avoid that lever from moving around. Not that you're going too fast, just make sure.
Oh, nice fish.
Just caught
Looks at frontless,
fish in the
right?
sit cam.
Oh,
Looks like it's
higher. That was lower.
so our sit cam and our side cam have slightly different angles. So the
There it
pilots
is.
are tracking it. So Alex has a buddy for his commute to the next experimental site.
He's unafraid. Some floods. Nice. Okay, you are further away from the vessel than you want to be. Just, yeah, look,
We can look around while you're waiting for the ship if you like. It's fine.
a small tip for you.
What's that?
Put out dial a little bit of downs, some clockwise here, where it's neutral. Then you'll be more comfortable taking your hand off, just doing camera settings and stuff.
Yeah. Something about the glowing one was going up.
I'm sorry, can I didn't ask.
No, so you can move it. The thing is like if we see something, it's actually better to center the camera and let us fly to
Yeah.
good view.
Yeah, so I was aiming at the shrimp right there.
Yeah. So if you want to see the shrimp, just let us know.
Yeah, we'll do that.
Because we can get you real close, and if you want to like zoom in and dial it in once we get there, that's fine. Or obviously,
Okay.
that's fine.
Good.
So right now, Falcor 2 is moving south to accommodate that move for Sebastian, but we don't want to get too far away from the ship.
It's this guy here.
Which one?
Taking a little bit of time to
Yeah, that's a species of sea cucumber. You have one here, one there.
Explored
Probably
seafloor.
some species.
I don't think I've ever seen that small.
The thing is, we never get that close. Often,
this line of stuff drifting across the screen.
when we are here, I mean, when we're flying around, we rarely stop to look at things. So, here,
Yeah,
while
once
we're
it
waiting for the
attached it.
ship, this was an opportunity to do it.
Seems like whenever we do a pause in a place and just look around, we always find interesting things down here.
Yeah, you know, that's the crazy thing. Like you showed that aft camera, right? And you're showing how fast life dies out, and it does. But then you go over to there and like zoom in, and you're like, oh my god, there's actually a lot of stuff here. It's just not really visible, and just different species altogether.
Yeah, it's a great reminder. And last expedition, when we do those heat flow events, we'd be sitting down for 30 minutes on what seemed like a barren seafloor. Just get the camera and you'll find all kinds of stuff down there.
See that sea cucumber again in the middle.
Show your floods.
We had roughly 2 hours.
Those colors look nicer.
We have bishop spiders, so we're going to buy an eye and probably a piece of chili today.
Did you slurp and everything?
Did you just flip the intercathodics on?
I did, yeah.
And everything that was on,
No,
on
just,
the list.
Hmm?
We got the rock for Avery as well from the bottom.
But not our collection site.
Not at, sorry?
Not at our collection site.
Yes. What do you mean our collection site? That's an experiment one.
Yeah.
Yeah.
This is not for her.
Ah, it's not?
Mm-mm.
Okay, so she wants what? You wanted a piece of rock there as well?
So what did you do after completely scraping off the bottom?
We used the scoop to get some of the rubble at the bottom.
Yeah, and this is for me.
And this is for you?
Yeah.
Okay, okay, okay. So
And for her,
and there's a bigger piece like this.
then she gets a little, so I have to look at it then.
We can
Because
also,
she wants like more, she wants more, I don't know what she wants, more rubber stuff, she has to break it.
well, she was very happy with what she saw in the scoop as she was in here. So
But where's the rock then? That also at the bottom, or what?
so
Like
you
this
want a
inner-
bigger piece? You want a bigger piece
No,
from the bottom?
but where's the rock collected actually for her?
At the bottom of the hole.
Okay,
In
then
the
she has
rubble
to
there.
wait there and look at this because
But we also have plenty of time. So if you want me to go back there and get more for you,
no, the rubble is here also for me, no?
it's also for you. So if you
But
want,
if
do you want
there's-
more
I don't know. No, I don't
for her or?
No, she can have it after me, but I look first.
Okay.
Okay, that we do.
I think so. Yeah.
Yeah.
Okay. That was not. Yeah.
Okay. This is full so far, but we don't know if it.
It's not moving. I'm keeping an eye on
now.
it.
Yeah.
So it's a pretty good sign. So
So They must be all full then, no? You have everything full from the suction sampler then, no?
We have still a fine and ultra fine. Sorry, 2 fine and 1 ultra fine ready for Bishop Spire, and we had 1 extra, just in case.
So 3 done, the 4th done, then 3 for Bishop Spire,
Yep,
and 1 is extra. Yeah.
yep, that's right.
Just 2.
No, roughly we have to be at Bishop Spire in 2 hours and 45 minutes, so I thought a little trip to Bio9 will be okay.
Yeah.
Grab a piece of the chimney at this time, measure temperature, grab a piece of chimney, and
And then
then,
this I mean, yeah.
yeah. And do you want to get more from the bottom then? More rubble and rock, or?
How much rubble and rock do you have? Not the rock, yes, but the rubble.
The rock is like big, and rubble there were like a dozen pieces or so. Do you want to get more? We can.
Yeah, we can go back.
Yeah,
I mean, no,
it's-
not now.
Yeah, of
So
course.
basically, let me look at it.
Yeah, so we're gonna go for the chimney piece right now. It's not gonna take very long, and we'll still
No, no,
have some
this is
extra
okay.
time.
It's
so-
all okay. If I want more, the hole is still there.
Yeah, absolutely, absolutely, yeah.
Because tomorrow we will go there also.
That's talking about me, so.
Yeah, so Monica would like to look at the rocks before you grind them.
Sure, yeah. And are
Um,
you guys getting back on the salt here or no?
no, we're going to log it now.
Okay.
Quite
Gotcha. Okay, sounds good.
Put a piece of chimney into a quiver.
Yeah, that'd be great.
Yeah, but if we need to put something in there.
You want some drone?
Is it- is-
Do you want some drone?
I don't, there was nothing planned in that file.
I want to say we
That was-
were there about 22.
Yeah, that's fine. Okay, yeah. It would be nicer from the same side.
It's got a couple different camera views up here. So you got the sit cam is the picture-in-picture, and the side cam
So you see the
is the main camera view.
feed, watch and recovery.
And so often
We usually try to
the
go
pilots will
around
use
first.
both views while they're doing these transits. So they'll keep their eye on what's going on below the sub, do a different view with the side cam. So it just gives them a better quick reference idea of what's around them, especially when they're in dynamic environments like this.
This is gonna take a while. Bridge RV, sorry about this. Can we bump the speed up a little bit? How about 0.3, 0.4, somewhere around there?
Yeah, call me out, right?
I got lowers on already. He's just so transparent.
It's like Alex has found something here in the water column.
Oh, I bet. If I turn those off. There you go. That one.
Now what is this
If
guy?
I do that. Yeah, and it's looking at the background. Okay. Maybe give you a touch of zoom, or you? Yeah, I'm pretty give you a little zoom. You want to creep that up.
So while waiting for Falcor 2 to catch up to the sub,
I heard of oh my god.
we're taking a
I was not supposed to outside.
little break to set up a shot that may look more like some of our mid-water shots, but giving a little something here on our transit to Bio 9, which is the site we're heading to next.
Thank you.
Why? Why? Okay.
Now the ship's caught up with us and put a little bit more speed on, so we're gonna keep moving south. And on this transit, we're getting an eyeful of the stark and sublime features down here and some really new crust.
So for the rising sponge there, these sponges actually are carnivorous sponges, but as you can see, sometimes there are some amphipods associated with them as well. Crawling at the surface there. No, it's okay. Just a good way to spend a few minutes. Oh, that's good.
Going through the other day. Or actually, no, that was up north. Yeah.
It just looks big.
Yep.
Doesn't seem like somewhere you should be allowed to fly as responsible. It's something from any
There's a crinoid there. And a little below that we had a little brachipod as well. There. I think this is a brachipod, I'm not sure. I don't know if the shell is hard or not. We'll see. Beautiful crinoids.
It's there.
But it's not.
Ah, you want to know when it's
There's a little smoother and easier. Pull everything off.
Oh, right.
Although more reactive, but
Yeah. No, that's good.
the It starts 16
Looks like we're about halfway to our transit to Bio 9.
I don't know what time was here. 16, 11, 4, then 1 hour and 45 minutes, if I remember correctly. And then 1 hour and 45 minutes after this. So, and that. That doesn't make sense. It's that like. Do you remember when we start walking with the.
Which one?
To the small
The
band?
little one.
1611.
And the ending.
Yeah, and they always band
And ended 1756.
You already have that to there anyway. Just in case. Yeah, so see how it's actually looking down? Yeah. So you easily like come into something that is pocket.
Oh, that's the one here.
I thought we went north.
Don't. I thought you grabbed that, looked a whole lot like it.
Yeah, you done was also just like.
As we navigate through this fractured landscape. These features are, geologically speaking, brand new. There's volcanic activity in this area, and we've been told that it's actually overdue for an eruption. I think it said it happens about every 10 or 11 years, 12 years.
Yeah, you can see all the sediment and suspension in the water.
So that's a sign of getting closer, Stefan?
Yes, it is.
Are we ready to be stopping here, mate?
Yes, sir.
Copy that.
Yeah, I can find two. You're still under 100. Pretty much there. He's got a ways to go. I think it's the alright.
So as
There
we're
we
seeing
are.
more,
Off to the right.
yeah, sediment on the surface and sediment in the water column, and here we are.
Pan up to him.
Yeah,
There's
thick
cyanide.
black smoke,
It looks like pea vent.
some spires in the distance.
Hey, could you get Stefan?
Yes.
Looks like pea vent and not bioline there.
Was,
Am I confused?
was 737.
This is the best. It doesn't get better than. For our
You want
He's smiling.
Somebody put a mic on this guy.
Black
Bears
smoker is
mic'd
always
up.
so dramatic. The life in front of the black smoke.
Let's go check out the smoker, right?
Yeah, this is a stunning view.
Do some like wraparounds.
It's already dispersing now. The life part.
Oh yeah, it's gone.
So is this structure built entirely from the minerals in the fluid flow?
Yeah, so when the hot fluid comes into contact with deep sea cold water, minerals that are contained in that fluid will precipitate. That's the black smoke you see. And through time, you will just accumulate a lot of these minerals. So that this plume, we call it, will rise into the water column and will rain over a fairly wide area. And it will also build chimneys. These chimneys will eventually collapse on the sides, become extinct, and they will accumulate large amounts of metal sulfides on the bottom. It's very strange because this looks more like P-vent than Bio9. I'm very confused.
I don't think we're gonna get you some lures. And let's decrease.
Yeah. No, decrease, yeah.
Chill. Can you zoom out a little bit on the map to see where P-vent is compared to us? P-vent is very close. Okay, I think this is P-vent actually.
Oh, it's very close to zoom in.
So what's the distance there? Because this looks like P-vent and not Bio9.
Can't find him. I'll go upstairs.
Okay, thank you. Do we have an upside down mirror? Not really. So these structures are strange, and they are formed when hot water is trapped under a flange, and they will eventually build an edge like this. And sometimes we have such a sharp difference of density that you have something that looks like a mirror underneath the flanges.
From inside, it's in the quality of the text.
Yeah, try to use the iris if you can.
There's still some fluid under there, I think. It looks darker.
Yeah, I'm stable.
Okay.
Pretty amazing.
So cool. There's a scale worm.
Yep, I know. Get it. Get it from me, please.
Just grab it with the fingers.
Exactly. Delicately and put it into the biobox. Good. Okay. I have to make a decision here. So a sample from P-vent will be okay as well?
Oh, totally, yeah.
Okay. Okay, so we have more choices here.
Okay, so what we're doing?
So we're gonna grab a measure temperature on the black smoker and grab a piece of it. So we can start by grabbing to make sure that's the one that's gonna be convenient for us, and then we'll measure temperature there. That was pretty amazing structure there, right?
Yeah. So I guess we just get a temperature about now. If I can push out forward too. So that the camera views.
So here at P-vent, the structures tend to be pretty tall, so we'll have to find a place where you guys can work. And there are plenty of different smokers, so we'll have to pick the one that is more convenient for you.
So you just need us to target, take the temperature first off.
So if you're just joining us,
Yeah, it can be either temperature first and then the chimney.
this is the Underworld of Hydrothermal Vents expedition, and you are seeing live footage from ROV Sebastian, Schmidt Ocean Institute's remotely operated vehicle, and we are here live in the mission control room on research vessel Falcor 2. So we've just transited to a new site. So our previous site had some lower temperature venting and some incredible science taking place there. And here we have some higher temperature black smoker venting and some visually dynamic views of vents down here on the seafloor.
Hey, so we overshot and we are at Pivent. Yeah, I mean, Bio9 is very close by. I think it might be better. I think there were more black smokers and more communities.
At Pivent.
At Bio9.
Pivent is like littered with chimneys there. You'll
Okay,
see.
we can check it
Yeah,
out.
have a look because we just grabbed a T probe now, so we're waiting. We're looking for you to try to.
Yeah.
Because I got very confused when we got here, and then I was pretty sure it was Pivent. You'll see. It's a nice picture, isn't it?
So we got Dr.
All the
Stefan
chimneys in the
Ordez
background there. There's the small one with the T probe
on the science mic, and he's conferring with Dr. Stefan Sievert
Yeah.
to determine what black smoker vent site
This one you're unlikely
they would like
get
to sample.
this-
Yeah, no, exactly. Yeah.
But maybe the one behind.
I think we are at Bio9.
No. By now it's like a single chimney.
In the
right?
meantime, just
No,
enjoy the
true.
view.
Okay.
Pivent is single chimney. That's Bio9, and there's Bio9 and Bio9 Prime, so like a north and south structure.
Okay. So.
Yeah, so yeah, actually Bio9.
You've already been in here?
Yeah.
You want to talk about that?
I'll seem to be doing just fine.
So, can you, do you guys?
We could measure the temperature before, but we could also measure the temperature after we've taken the piece.
That'd be best because they had me pick up the probes.
Okay, sure, yeah. So I think it's best to find a place where we can be stable and have good access, and not and to shoot for not like a super big structure, but like I don't know, something like this here. But that might be a little hard to get to, right? I mean, we could sort of do a survey of the area and find out like what the best. And there's also additional structure, just like I think slightly to the north of it, or to the south. I'm not sure. Yeah.
So we got Dr.
Yeah, this over here.
Stefan Sievert is in the science chair. Still Alex piloting. Jewel co-piloting. This is Tyler, one of the marine techs. And I will be vacating the seat here in a minute. But the team is looking for an ideal place to park close to the vents so they can do some sampling.
I think so.
So if we switch to the sit cam, in the bottom right hand corner you can see the starboard ram. So landing out on a structure like this isn't easy. So the team's going to look for a spot where they can delicately set one or both rams against the vent, and with the minimum amount of fuss be able to reach the sampling site. But as you can see, these aren't large, they're not flat, They're not regular shaped, so it's going to take a little bit of planning to find a good spot. Also, they can be crumbly as well, so take a bit of deft pilot work to get settled out here.
Let's go higher on this one, see what we get.
That might be hard to position yourself, right?
We gotta look first.
Yeah.
So right now they're doing a bit of reconnaissance.
That's tiny smoke right there. It's so cool.
Choo choo.
Look at that guy. That looks almost man-made.
Yeah, it's like a spear. So you can tell some of these chimneys
I would,
can grow quite tall.
if you're wanting to do that, I would rotate around the first one and come in this way. That way everything's in view the whole time. But to
And
just
that
try to do
thick
it,
black plume
I would not worry.
is also lowering visibility a bit here. So whereas previously today we were at a venting site where the vents are cracks on the seafloor and the vent fluid is clear and you see a little bit of shimmer in it, these are the black smokers, and they have earned that name pretty clearly.
Smoke on this side. Fine. Should be it, I believe. Come in,
I don't want to smash it.
So if you notice this thick smoke-like substance we're flying through, this is some mineral-rich fluid that's ejected from the vents, and as it settles out, it builds these chimney structures that we're seeing on the seafloor. So in a way, the vents are building themselves. They're building their own chimneys.
Mostly south to get it.
yeah, follow the cloud down. There's the
So as a bit earlier in a transit, we saw some seafloor that was cracked lava. And I'm not a geologist, so forgive me if my descriptions aren't 100%. But this is a different material, a different look, and a dramatic look.
Star one or something maybe? But this is the first one we came up to.
Yeah, I think so. Maybe go.
I liked that spot we were at first, where you could put your starboard ram against.
Like here.
To the right, put the ram a little to the right, probably.
Yeah.
Yeah, you're gonna want it on the big base there. You can work on that one and all these kind of. at the same
Yeah.
time.
Okay.
You want to bring your arm. Are you happy with it?
Yeah, I want to bring it in. I was just trying to do your best for you.
Yeah.
Now. if you come in a little bit more of an angle, you'll be even safer. Running there, I think you're good. Visibility is not the best. You're going to be pulling that because you're flying forward. Never mind. Let's.
So in this camera view, you can get a pretty easy understanding of the difficulties of working around these vents.
So maybe on the other side of that. But hopefully the current will help us there. So, I wish I could help you with this video. I'm blocking.
Just have to be careful. Just be careful here. There's a temperature logger in here, so.
Yeah. So we explored this side of it, which is actually the worst side for us to work. That's the current. Let's explore this side of it and see if there's anything that's workable.
Yeah.
All right, so as the pilots work on landing out on this hydrothermal vent, I'm going to take my leave, work on some other tasks here on Falcor 2, and so we'll have Ivan Rodriguez Ramirez doing some narration for you. So enjoy.
You're shooting for these here?
Yeah, so kind of.
But
no.
that.
Oh, sorry. Go ahead.
No, these would be difficult to sample because we want to have like a chimney piece. So I think it's.
What I'm hoping for is if you land right there, there's a smaller one up higher. Hoping that that one in it has a column. So hopefully there's something for me to scratch. That's the one that you guys pointed out that we could work on from the first side. So it's the same one, just a different angle.
This one here, you mean?
No.
Up here?
Yes.
Yeah, this one would be perfect, yes.
Hola a todos, saludos desde el Falcor 2 y bienvenidos a esta transmisión en vivo. Gracias a las cámaras que tiene el SOI Divestream del Schmidt Ocean Institute. Yo soy Iván Rodríguez Ramírez y voy a estar acá narrando la siguiente hora de este live stream. La narración va a ser en español, así que saludo a todas las personas de habla hispana que estén acompañándonos por acá. En este momento lo que estamos viendo es el ROV posicionándose para poder colectar un segmento de estas chimeneas que vemos en pantalla. Hi everyone. For the non-Spanish speaking folks, I am Iván Rodríguez Ramírez from the University of Costa Rica, and I am going to be narrating the live for the next hour. The narration is going to be in Spanish, but you can always send your comments and messages in the chat in English, Spanish, or in whichever language you feel more comfortable with.
I mean, the question is also if you can then reach it with the arm to sample it.
The only way I'll know is if I reach out there.
Yeah, no, sure, of course, yeah.
Lo que se quiere tomar con esta muestra es temperatura y parte de la roca, de la estructura que forma la chimenea en sí. Podemos hacerlo de varias maneras diferentes. Podemos tomar primero la temperatura y después colectar la roca, o podemos hacerlo al revés.
Like over here, maybe checking this
Estamos
area
viendo cuál es la mejor manera.
Yeah, we can try, but I thought you said that temp probe did not.
That's over here, the temperature probe. So I'm thinking about
En este momento, en el micrófono, el investigador principal en la sala de controles es Stefan Schwieber del Instituto HUI en Estados Unidos.
So I'm thinking about like going around here and then maybe.
We can try. The problem is that's the backside of the current.
Él está dándole las indicaciones a los pilotos y conversando con ellos para ver cuál es la mejor manera para hacer esta colecta. Hay una pregunta en el chat de cómo es que se forman estas estructuras. Y justamente antes de yo venir para acá, estaba conversando con la estudiante que va a analizar la muestra y ella me contaba que la estructura se forma por la deposición de los minerales que están saliendo del agua de la chimenea. Resulta que este flujo de agua está lleno de diferentes minerales que están súper calientes. Y cuando el agua caliente choca con el agua fría, entonces los minerales se precipitan y se van acumulando, formando esas estructuras. También estos minerales son los que le dan el color negro al flujo que vemos. Justamente por esta razón es que estas estructuras son tan débiles y hay que tener mucho cuidado al momento de colectarlas porque se corre el riesgo de colapsar toda la estructura. So to answer an English question that was in the chat, the formation that we see right now is actually precipitated material that comes through the water flow, the black water flow that we see that is coming out from the chimney. So when the minerals, metals and other stuff precipitated, they accumulate and form this chimney that we're seeing. And that happens because of the difference in temperature between the water flow and the cold water that is surrounding it.
Can you click your frame grab or frame highlight?
Justamente le estaba preguntando a la misma estudiante Avery qué era lo blanco que está alrededor de la roca y me decía que ella cree que son justo mantos de bacterias que hacen quimiosíntesis y entonces crecen en estas zonas.
Lost my depth perception of the snow on this one. Let's see if I can get a second shot good. I know I created the snow level.
El ROV Sebastian tiene como este brazo metálico que se puede ver en pantalla del lado izquierdo en la parte inferior, justo detrás de donde están las lecturas de temperatura y de salinidad, y se quiere utilizar este brazo como apoyo, ponerlo en la estructura para poder estabilizar el ROV en una sola posición y lograr tomar la colecta. Se puede ver muy claro que la estructura es sumamente boronosa, podríamos decirle, porque con que solo el ROV lo toque salen muchísimas partículas.
I'm gonna try it. That's a little bit to the left. Wherever that
Pero acá
Can
los
you,
pilotos están haciendo su plan para poder tomar la colecta de una manera que no dañemos la estructura.
well, if you rotate, that's gonna bring that closer to us. Is that right? So I would maybe left,
slide
more
left,
than
yeah.
but at this point, like, let's try it because you're already in a good spot.
can you increase the light a little bit?
Yeah, just do
Great, thank you. Yeah, we can shoot for this one here. Okay, measuring, going to measure temperature.
Ya se va a empezar la medición de temperatura.
Can you get us a temperature overlay, please? Yeah. Thank you.
Maybe go a little deeper. Like right here. Yeah. Nice. I think that's it. Which one is the temperature? This one, or this one?
The one on top.
The one on top? Okay.
The one on the bottom is just the highest one for the past minute.
All right. So I would expect we should be able to measure higher temperatures. I'm not sure exactly we are in the right position. I mean, it's kind of narrow, but
You may want to break the tip, the top of the.
yeah. Now I think yeah.
So 83.2.
Yeah, I think it should be higher. But maybe it's better to take the piece first and then measure the temperature. Let's see. Now we are okay. Now it's going up. Okay, stay there.
Podemos ver lo caliente
Yeah.
que está el
It's very narrow,
flujo que sale
I think.
de esta ventila hidrotermal.
133, 130.
Esta
Yeah.
agua tan caliente llega a chocar con agua que está a casi 2 grados
be even higher.
centígrados de temperatura.
Yeah. Now we're talking. Okay.
And 78
Yeah.
max.
I would expect it to be even higher, but I'm not sure. Can we zoom in even more? No.
Acá el científico principal Stefan está diciendo que él
Yeah, maybe move a
esperaría
little bit.
tener una lectura de temperatura aún más alta de lo que tenemos en este momento. Acá podemos ver también la rapidez con la que sale el
think maybe back
flujo
here.
de la chimenea. Hay una pregunta en el chat sobre
Yeah, I think the hotter fluid is over here.
qué se
for
vio
sure.
en las
That's
muestras que
colder.
colectaron ayer.
Maybe over here.
Entonces, por si no nos pudieron acompañar, ayer se dio la primera colecta o se recogió el primer experimento
Yeah.
instalado previamente en unas ventilas hidrotermales para probar la hipótesis de las investigadoras principales de esta expedición, entre ellas Monica Bright de la Universidad de Viena. Y la hipótesis es de si larvas o microorganismos utilizan el flujo de ventilas hidrotermales como manera de movilización de un sitio a otro. Para poder validar esta hipótesis, ellas pusieron una especie de trampa que consistía en una caja hecha de una malla de 32 micras y la pusieron encima de una ventila hidrotermal de flujo difuso. La dejaron ahí por una semana y ayer se colectó. Además de la colecta del experimento, también se colectó un gran trozo de los basaltos que estaban debajo debajo de la caja. Y bueno, se encontraron varios tubos de gusanos debajo, en la parte de abajo de la roca que se colectó hasta bastante entrada la noche. La investigadora Mónica Bright estuvo con su microscopio analizando este trozo de roca. Todos estábamos muy emocionados a bordo. Era casi como ver una procesión, todo el equipo científico detrás de la roca. Y fue un momento de celebración, ya que esto validó básicamente la hipótesis que se tenía. También se encontró un poco de meiofauna no teñida. ¿Esto qué significa? Para también las personas que no han estado siguiendo en totalidad la expedición. Al momento de instalar el dispositivo, se pone un tinte. Con el dispositivo me refiero al experimento, a la caja que les acabo de mencionar. Se pone un tinte para poder teñir los animales que están en ese momento dentro de la caja, en el minuto 0. Y entonces al momento de retirar el experimento, los animales que se colecten y no estén teñidos, significa que entraron por medio de la ventila hidrotermal a esta caja de manera posterior a que se instaló el experimento. Y el hecho de haber encontrado un poco de meiofauna no teñida también implicaría que la hipótesis fue validada. Aún faltan 3 experimentos más, pero todos acá estamos muy contentos con los resultados preliminares que hasta el momento se han encontrado. Ayer lastimosamente se perdió la muestra de agua que se tomó antes de eliminar el experimento. Lo que pasó fue que justo antes de quitar la caja se perforó la malla para poder absorber todo el agua que estaba adentro y tomar la muestra. En esa muestra había bastantes bacterias, pero probablemente mucha meiofauna también. Pero por fallas mecánicas la bomba de agua quedó abierta y la muestra empezó a bombearse para fuera de la bolsa en la que estaba colectada, entonces se perdió. Aún así se habían tomado otras muestras de succión en la que se vio que hay una cantidad increíble de bacterias que si se van para atrás al inicio de este live van a poder ver grabaciones de los mantos de bacterias que están cubriendo el experimento. En la parte de adentro también hay muchísimas bacterias y esas sí se pudieron colectar, también con un poco del agua que está adentro, pero bueno, no lo mismo que la muestra inicial, ¿verdad? Pero aún así están las motivaciones bien altas, ya que tenemos otros 2 experimentos, entonces todo bien, pero sí una pena.
How do we do it? I'll follow you in.
If you want to run the camera,
Yeah.
Yes.
You grab that and then I'll.
There's people in the chat that want me to also narrate in English, so I'm gonna do that.
You want to unhook that first? Or do that?
I was just telling that we lost a little bit of the sample from the first experiment yesterday, which was a shame, but we still have other 2 replicates of the experiment. So everyone is very positive about that. Also, yesterday we collected a big piece of the basalt where the first experiment was deployed.
Do we weigh it? Those questions yesterday, how much we thought it was going to weigh. Did we weigh it?
No, not yet. But it was very heavy. Acá uno de los pilotos me preguntó que si pesamos la roca. Y no, todavía no la hemos pesado, pero era un trozo muy grande de roca de basalto y era sumamente pesado. Varias personas tuvieron que ayudar a mover la muestra y llevarla a los laboratorios para que fuese analizada. As I was saying, yesterday they took a big sample of a big piece of basalt. It was the piece of basalt where the first experiment was deployed, and they found, and you can see in the live from yesterday, that there were little tube worms inside of the rock, which was very, very exciting. And then one of the pilots have just asked me if we weighted the rock, because it was very big and very heavy, but we haven't done that yet.
You want to nudge that probe over?
Be careful, there's starting to leak on there.
No, that'll be fine. That'll be. How about steamy? See that? All clear. We'll figure that out when we get to that. Right.
You do a highlight?
Thank you.
Alright, you see where it's on the black one where it's got the little spin part
Okay, so most likely I'll go back to the side.
Highlights.
You gotta rotate your rail in. Probably the easiest way to do it. Stay by though, maybe
De hecho que conectando con esto de que la muestra de roca de ayer fue sumamente pesada y viendo la especie como de pala que tiene en este momento el ROV, cuando uno ve estas grabaciones parece que todo es muy fácil, que lo que estamos viendo son solo pues, herramientas, pero en realidad este instrumento que estamos viendo en pantalla es sumamente pesado. Uno lo puede alzar con una mano, pero es más pesado de lo que parece. Y lo que pasa es que los brazos que tiene el ROV son 2 brazos industriales que pueden levantar hasta 200 kilogramos de peso. Entonces, si ven el live de ayer, que les recomiendo que lo hagan, al momento de levantar la piedra, el ROV lo puede levantar como si nada. El brazo lo levanta con bastante facilidad y se hace con la colecta con facilidad. Fue un momento sumamente tenso dentro de la sala de control, pero se logró hacer con éxito. y el ROV pudo levantar esta roca con un solo brazo. Y luego en superficie en el barco se necesitaron como 4 personas para poder levantar la roca. En los comentarios hay una pregunta de cuál va a ser el futuro de esta roca. Y justamente ayer los científicos estaban conversando toda la logística de exportación para poder llevar este pedazo de basalto de vuelta con la investigadora principal a la Universidad de Viena. I was just saying that when we see the ROV manipulate the different tools that it has, it looks pretty easy, or at least they look pretty light. But in real life they are quite heavy. And that reminds us that the ROV arms are very, very strong. They can lift weights of almost 200 kilograms.
Go down a little bit and get some more.
And if you see the light from yesterday, when they collect the big piece of basalt, you can see that they did it just with one of the arms of the ROV. And when the big rock came to surface on board, there was like 4 people trying to move it to put the rock inside the labs.
Okay, let's. Yeah, I think that looks great. Yeah.
Okay.
Let's get that.
Let me open that for you, or you okay?
I'm just gonna hold this here, just what I'm gonna do actually, I'm gonna pass it up to you, and I can do the.
You want to just put it in the. What will we put in the bio box? Okay.
It's just easier if I can control the fireworks. I'll zoom out.
105.
Camera's coming back in.
What sample number was this?
105.
I'm gonna double check.
Acá ya se logró conectar
Firebox number 3, right?
el segmento de la chimenea. Como podemos ver, la estructura no se ve afectada.
And
105.
That's the chunk there.
Okay. Nice work. Yeah.
I don't think so. Yeah.
Okay. Close the box and then.
Okay, that's it.
Anything else you want to collect with the scoop before I put it later?
No, I think it's about.
That's it. Next
Hmm?
one will be suction samples.
Hmm?
Have to lift it out.
The next sample will be suction sample.
Yeah,
You don't need
right.
to-
Yeah.
Scoop? All right.
Do you want to draw that front one, do the back one? Yeah, good.
Yo viene el Nautilus Max. Nautilus, yo me meto en el Nautilus, claro. All the way out? Not yet.
As far as the wall. Oh, I am sorry, the wall.
Acá los pilotos le preguntaron a Stefan, el investigador principal, si iba a necesitar otras muestras con esta herramienta o si la guardaban de una vez. Y se tiene que preguntar esto porque el tomar la herramienta, acomodar el brazo, acomodar el R&B toma bastante tiempo y el tiempo que tenemos en el fondo es limitado, por lo que se debe de ser lo más eficiente posible.
I should close it, please.
It's almost something.
I find it.
Nice.
Sitting just
Totally,
right.
totally meant to do that. On purpose.
Acá el piloto acaba de bromear porque la manera en que guardó esta herramienta, esta palita, fue bastante sencilla, se vio.
Open it up.
Do you need to leave at 3 to be back at 5:30?
Y entonces nos decía, claro, yo lo iba a hacer así a propósito. Y todos nos reímos porque fue muy gracioso. Normalmente, como les decía, el guardar y el sacar estas herramientas toma su tiempo. Y con solo abrirla se acomodó perfecto.
Like where we just open the orifice to basically go back
Yes, fine.
and just measure it here.
Just take a few minutes. for this.
Okay, that would be great. That would be awesome. Yeah.
Just put the bungee back on this.
Yes. Okay.
So you don't want to take any more samples here, no?
No,
No. That's it.
that's it. And just measure temperature one more time, and then we're done. Here.
Stefan,
Yes.
there's a question in the chat. Do you have time?
Yes, of course. Yeah.
They wanted to know, how do you know that you have enough sample?
There's never going to be enough. We'll try to get as much as possible, but also we fully recognize that it's we have to take get what we can, or we have to take what we can get. And so in this case now, it was very difficult to get this piece of chimney. So whatever we have to work with. Ideally, we would like to have more, but it's already great to have that piece. It's very difficult to get that kind of sample.
Thanks. Stefan nos estaba diciendo, bueno, la respuesta a una pregunta en el chat, que preguntan que cómo se sabe que se tiene suficiente muestra, porque parece que lo que se muestra fue muy poco. Y Stefan nos estaba diciendo que en realidad uno siempre quisiera tener mucha más muestra, ¿verdad? Para poder analizar más cosas, hacer más exámenes, pero que tomar esas muestras es algo complicado. Entonces, solo usted es agradecido con lo que se tiene. En este momento, esa cantidad fue lo que se pudo colectar sin dañar la estructura, sin dañar la estructura de la chimenea en este momento. Así que solo se acoplan los experimentos y los diferentes análisis que se le vaya a hacer con la cantidad que se tiene.
Is my PhD advisor. I'm going to be processing the chimney sample. And my idea is to process, basically to subdivide it into 3 different samples and then do 3 different analyses with it. And in this case, I mean, we always say in science that we're, sort of resting on the shoulders of giants, and to determine the amount of cells that I need. It's a very little amount for microbiology, but the way that I determine that is actually by going into the scientific literature and reading about how many cells are needed for the specific analyses that I'm going to be doing. For these rock samples, I'm doing rock samples both from the seafloor and from these chimney samples, so I'm expecting to have a lot more cells in the chimney samples. So even a little bit of a chimney sample will be enough for my analysis, versus for the rock samples I'm going to want around 10 grams per analysis that I'm going to be doing. So yeah, that's a little bit of insight. Feel free to ask any questions.
Muchas gracias a Avery por contarnos un poco más y ampliarnos más el panorama. Ella nos decía que para ella poder saber cuánto va a necesitar para sus análisis, recurre a la literatura que ya hay, y ella va a comparar con comunidades de microorganismos dentro de las rocas que construyen estas chimeneas, y también de los basaltos o de las rocas que están presentes en el fondo en las ventilas de flujo difuso. Y nos decía que ella espera tener muchas más células en estas rocas de las chimeneas que vemos en este momento que en la de los basaltos. Y que necesita como 10 gramos de muestra de basaltos para tener la cantidad de células necesarias. Por lo que la muestra que tomamos ahorita probablemente sí sea suficiente para poder colectar las células que están dentro. Avery ahora nos va a responder unas preguntas para ella que surgieron en el chat.
Hey, so I'm actually not a geologist, I'm a microbiologist, but I'm working with both rock samples and water samples, so we're really interested in the samples of basically bacteria and archaea, so single-celled basal organisms that are occurring both inside the rocks, on the surface of the rocks, from diffuse flow vent fluid, and from the overlying seawater. And the rock samples that I'm looking at are both made of basalt, which would be the sort of samples that are coming from the seafloor and from these diffuse flow vents. But when you have the black smokers, basically they are so concentrated in vent fluid, they haven't mixed basically under the seafloor with the overlying seawater, and so they're really concentrated with all of these minerals. So when they come out of a very specific crack in the seawater and you have this really focused flow, you have very, very hot water, about 250 to 300 degrees Celsius, that's mixing with background seawater that is 1 or 2 degrees. And so you have some of this like instantaneous precipitation of these minerals. So most of the chimney is made of different minerals such as sulfide and calcite. So that's what the chimney itself is made out of.
Avery nos estaba respondiendo una pregunta en el chat, que la pregunta, bueno, las preguntas eran varias. La primera era que si ella es geóloga por estar trabajando con rocas, y la respuesta es que no. Avery es microbióloga. Y la segunda pregunta era de qué estaban hechas las rocas, si están hechas de basaltos. Y bueno, acá Avery nos dio una respuesta un poco más amplia y nos contaba que ella es microbióloga y trabaja con rocas porque estudia las comunidades microbianas que viven dentro de las rocas y también en la superficie de estas. Ella trabaja no solo con rocas, sino que también trabaja con agua, muestras de agua, viendo las comunidades también microbianas que se pueden presentar en esta. Y con respecto a si todas las muestras que ella trata son solo basaltos, pues en realidad no tanto. Una de las muestras sí son basaltos, pero otras muestras como las de estas chimeneas son sulfatos y calcita. Esto porque el flujo de las ventilas hidrotermales acá en estas chimeneas es sumamente concentrado cuando estamos hablando de minerales y metales. Y como les comentaba anteriormente, como este flujo es sumamente caliente, al chocar con el agua súper fría, se da esta precipitación casi instantánea, y se acumula el material y se crea la chimenea. Entonces Avery nos estaba contando que los materiales que se depositan son más que todo sulfatos y calcita. Así que eso es lo que ella va a analizar de la muestra, además de los basaltos que se han colectado en las ventilas hidrotermales de flujo difuso en inmersiones anteriores. De ROV, entonces vamos a hablar con el piloto que tengo a mi izquierda, Chris, y nos va a contar cómo es que alguien puede convertirse en un piloto de ROV. ROV pilots. So let's talk with one of the pilots. It's called Chris, and he's going to tell us a little bit about how did he become a pilot. So Chris, how did you become an ROV pilot? And how can someone that actually wants to become one can do that?
All the pilots that are out here now kind of all came from different places, but with the same basic concept. For example, I'm former military, and in the military, I went to school to study electronics and other things in the Navy NECO program, and that background gave me an understanding of electrical theory and application, and so that was the foundation for what I do. And then just by random chance, I knew somebody who was running a job using an electric ROV doing inspections after Katrina, and he asked me if I could help out just because they were running behind. So I went out just as a favor to a friend, and that was a long time ago, and I liked it, and enjoyed the people I worked with and the places that I was going and the work we were doing. So I decided to keep doing it, and then just learned the other aspects of it, the fiber optics, the hydraulic theory, things like that. Learned how to fly an ROV, learned how to use the manipulators, everything that's involved with being a pilot. I learned that from people that I worked with, taking courses here or there. Just picking up what I could when I could. And I know that a lot of other people, there's schools that you can go to We have a couple guys out here who've gone to ROV schools. They teach you the foundations of what's needed, because out here we're kind of a one-stop shop. Everybody who flies the ROV or works in the manipulators also works on the ROV, fixes it, preps it, installs everything that needs to be on there. I find a lot of different companies who are working. So that's how it works for me.
Great, thank you. Chris nos estaba contando un poco sobre su experiencia, cómo fue que él se convirtió en un piloto del ROV y cómo personas que estén interesadas podrían convertirse también en pilotos del ROV. Chris nos contaba que, bueno, los pilotos que trabajan acá en el Schmidt Ocean, específicamente acá en el Falcor 2, vienen de diferentes caminos de la vida, y que él específicamente viene de la electrónica y empezó a aprender más y a meterse más dentro de todo este mundo de los ROVs. Dice que se tuvo que preparar y aprender más sobre los temas de fibra óptica y oceanografía, pero más que todo en la parte de ingeniería, de hidráulica. Luego de esto tomó varios cursos para aprender a manejar ROVs, cursos por allá y por acá, como dice él, ¿verdad? No en una escuela específica, pero que sí hay escuelas que enseñan el manejo de los ROVs. Que de hecho algunos de los pilotos que trabajan acá a bordo del Falcor 2 han ido a estas escuelas para poder aprender. Entonces dice que si alguien está interesado, sería bueno que se ponga a estudiar dentro de estos campos, más que todo ingeniería, fuerzas hidráulicas, fibra óptica, porque al menos acá a bordo del Falcor 2 los pilotos también hacen otras cosas. Tienen un puesto bastante integral en el que no solo manejan el ROV, sino que también le dan mantenimiento y arreglan lo que se tenga que arreglar, y son los mecánicos o los ingenieros técnicos de en sí el dispositivo que es. Algo bueno es buscar empresas como esta en la que él trae la oportunidad de manejar posado encima de las rocas.
Is that a little stock crinoid on the next little wall over? There's a tiny thing.
Oh, can I zoom?
Sorry. You're afraid of before, but. Yeah, no, it's not. Could be. Well. Yeah, it happened to me. Go ahead.
Aquí en pantallazo blanco que vemos es una langosta. En este momento nos estamos movilizando a otro de los sitios de interés en la expedición. Este punto se llama Bishop Spire. Y acá Chris me acaba de contar que está como a unos 60 metros. Entonces ya ahorita llegamos. En una de las expediciones anteriores el ROV también tenía que desplazarse, pero era una distancia un poco más larga, y eso requirió que el barco también se reposicionara. Justamente en esa expedición tuvimos otro problema con la muestra de agua que se toma en la bolsa. La bolsa se infló y se despegó de uno de sus lados. Entonces esa movilización se hizo muy lentamente, porque corríamos el riesgo de que la bolsa entrara a alguna de las turbinas del ROV y se dañara. Pero por dicha, los pilotos hicieron un excelente trabajo y eso no sucedió. así que no perdimos ni la turbina ni la muestra de agua.
Yeah, just kind of seeing that.
Have we been here before in this region?
Yeah, your kids' experiments there.
Yeah, these are children's experiments we checked out this morning. So in the foreground is the one on bare basalt. In the background over there, you see two more lids that mark the site, and these are obviously closer to an active site. And we're gonna now go to a very active spire as well, covered with small alvinella worms. There's recovery for tonight.
Acá marcan que era más apetecido por la fauna en los fondos. Entonces en esos experimentos se puso frijoles, carne, hígado y grasa para ver qué era lo que los animales preferirían comer. También acá a bordo tenemos a un artista que su nombre es Max Hooper, y él hizo unas esculturas que se bajaron con el ROV. Entonces también vamos a ir a verlas y probablemente también a recogerlas ya y traer la superficie de vuelta. Como podemos ver en este momento en pantalla, una cámara externa ubicada en el Farcourt 2 está lloviendo. Tenemos un tiempo no tan soleado como antes. Eso puede afectar un poco nuestra conexión, entonces si el live se ve interrumpido es justamente por el tiempo que tenemos en este momento, pero por ahora todo está bien, entonces acá seguimos.
Ready to accept that I'm not in the right spot. So this thing we pick up at the end of the class.
Are we ready to stop in here, guys?
Ok, creo que antes no se veía, pero ya se está viendo en pantalla la cámara externa del Falcon.
Yeah, Tony, you're in a good spot right there, so just whenever you can ease to a stop, it'd be great.
Que tenemos bastante lluvia hasta el momento, bastante nublado. Ahora en la mañana estaba bastante soleado, pero bueno, así es el tiempo. Acá lo que vemos en pantalla es la escultura del artista Max Hooper.
So first thing I'll do here is just document the art, and then we will measure temperature and do a little bit of suction sampling on the surface of the tuning.
Okay. With regards to temperature, we taking the same.
Entonces, acá con las estructuras de
We're probably gonna do something like there.
Nice scan.
Thank you.
I don't know.
I was wondering if you would show up.
Very steady.
Y bueno, con estas imágenes de la escultura de Max, yo me despido. Muchas gracias por acompañarnos en el live. Aún tenemos un poquito más de tiempo acá en el fondo. Así que no se desconecten, aquí seguimos y nos veremos mañana. So thank you guys. With these images of the Max's art, I'm gonna leave you now. So thank you for being here in this past hour in the Spanish section of the dive. So thank you, and I'll see you tomorrow.
Oh crap, God, there's so much, it's crazy. It's also, I mean, look how quickly the chains got.
Colonized?
Yeah. I think today, this is the best yet.
Looks like a Sharpie. Look at the skin, like loose.
Okay.
Yeah.
Dude, good call.
The plastic, right? Oh, the epoxy. Is
That's
it
gonna
epoxy?
be our bottle of wine conversation. Because I have a long-term thing in mind.
Thank you to Yvonne for narrating for the past hour. Some of y'all might notice that we do have some crummy weather out there. So if we're buffering, and I've got to imagine we probably are, a little patience, and it looks like maybe a bit of blue skies ahead.
It doesn't have as much tooth to it.
Probably.
I think the metal's got a little bit
More roughness, surface roughness that
Yeah.
Okay.
Thanks.
You're welcome. I like this as well.
Yeah. Thank you.
Okay, guys, we'll want to measure temperature, so you'll have to reposition, is that right? Or
Yeah, so what we'll do right now is we'll just do a quick check of the valves, make sure everything's off.
Yes,
Just a quick
sounds
square
good. We'll do this, absolutely right. The bag looks pretty good, but better safe than sorry. Oh, still zoomed in, right? I know, I'm all the way out. Okay.
It's good.
Down the center. Well, you can see it down right there. Yeah, it's over there.
Yeah, that's good. Okay.
Good.
Whose point we want to test first?
So-
That corner?
We can start here, but eventually we'll want to do 2 or 3 more spots to try to characterize a little bit the surface temperature. So we're not gonna pierce the thing. We want to see where little animals live at the surface, what the temperature typically is.
Temperature up on the screen, yeah.
So come to touch the surface, but not trying to get. Sorry, you can just anywhere in that area. Oops, sorry. Should have first centered. Are you touching or are you still far? Okay.
Center off, yeah.
It's about 12.6 degrees Celsius where it currently is. It was a little bit of variability. Chris, could you move it up maybe into that area where it looks like there's some shimmering, please?
You go where the shimmering is above it, or where?
Just like to come into contact with it around there. Well, it's perfect. Taking Alvinilla's temperature right now. So the gradients are very steep, so not very surprisingly, that's good there. Temperature is cooler than one would expect. So we went up to 8.9 earlier. It's pretty good. Alessandro. Hey. Six. 6.7. Okay, so
First check. How you feel? Nah, it's okay.
Maybe I zoom in on this side.
we'll move.
Okay.
Surface of this Alvinella colony, so if you can go around here, also on top is also fine.
Okay.
Wherever is easy. Yeah, there.
There's loads in there.
A bit. Yeah, I think that probably once more and then it's good. We have nothing yet.
Yeah. Let's turn that off and dump that one. Oh, it's gone. Okay, yeah, great. Oh yeah, just let it run for a second to clear all that stuff out. Shoot. Alright, looks like it's good.
It's good in there.
Yep, no, you're good. So, yeah.
What would you looking at?
That's good.
Are you happy? Happy with that?
Yes.
Okay.
Right.
Okay.
No more sections.
Yeah, we are thinking about, but this I need to ask Stefan because he's in the lead here. If we now go to back to the school experiment, like we have like about max 45 minutes left,
Okay.
and we need to recover the hanging man.
Do that now.
But if it's on, I think it's very difficult. It will be difficult to fly around.
Yeah, I think so too.
So I would like to discuss with him, what we do.
What is in the box? Is it what we got inside the cooler? You know,
We need to, need to wait for Stefan.
Oh, yeah. Yeah, this is what.
Nicole, can you go and look for Stefan? We want to see back in a minute if he's around. For this.
And we start heading in that general direction? We'll start heading in that general direction?
Yeah, I want to just wait for a moment, 1 minute, and he should be back soon. Because I was not here in the beginning.
Are you looking for Stefan?
Yes, and he should be back in a minute.
Yeah, he's just chatting with Monica.
Yeah. Ah, okay, there. That's why he was caught up.
Speaking of the devil.
Sorry, Monica and I were discussing about
Okay.
what we should do next.
Okay, so
You
this is
got
what I
what you
wanted to
needed?
ask you. So we are done here?
So you had that ultra fine and fine.
Fine.
Both?
All right. It looks like we might be slightly recovered from the weather. We've got that over at the side. But I think it's moving away. So fingers crossed. Thank you for your patience with our connection here. We currently have about 45 minutes left on bottom. And scientists are currently kind of debating in what order they want to achieve the rest of their objectives here in that time.
But for the suction sampling for you or for you,
The last jar we can do.
look, try to use the last jar what you like. And in the meantime, I ask Jan if he wants a scoop of mussels or if he wants a grab of, I mean, I don't know.
No, okay. Okay.
Because we are here. And
Yep.
then we throw this in the big bio box and he can do whatever.
So sorry, we'll need to grab the suction sampler again.
Sample number 106 is in jar 4.
6, yes, got that.
Sorry, is only 3.
And
107 is number 2.
107 is number 2.
So I think it's likely to test, but you couldn't see it because it was all like...
So we only have jar number 1 left, correct?
Yes.
Yep, that's it. So we're gonna suction around the base of some riftia around here. So I mean, we may have to relocate a little bit. And the last thing we'll do on this dive is just pick up little man there and grab it in the arm and hold it in the arm until the end of the dive. So let's not go too far. That's what I mean. But we have time. We're good on time. Oh, there. 2 worms are right there. So
Where are you checking for your stuff, or you just look around?
just looking around a little bit and just going to suck at the base of the rift, yes,
Okay.
I think, so that there could be
Few
limpets in there. Yeah.
Okay. So, wenn Scoops vermuscheln müsst, da irgendwo, weil wir bleiben jetzt irgendwie da bis zum Ende.
Want to put this one up?
Yeah. How many?
Die Biobox hier raus, ja.
That's behind us right now.
Well,
Nice view of the tower.
it would be great to get some of the associated fauna.
Suction Sample haben wir keine frei. Du musst das Ganze
So suction
nehmen und
sampler is
einreiben.
enough.
Aber es ist das Gleiche.
You don't need muscles, right? You don't need the fauna that is associated with the muscles.
I don't know who's getting the suction sample.
Suction Sample are full. You can
No,
take.
because you just said.
This is for
Okay, yeah, maybe, I don't know, 10, 15, something like that, so.
whole scoop.
Of what?
Yeah.
Scoop muscles.
Muscles,
Yeah.
okay.
Throwing muscles.
Okay.
I went here.
Sorry.
Muscles, yeah.
Maybe
Causing up.
even in the upper left, there is quite a lot of stuff on them.
Yep. So I will do a bit of suction sampling around here, and then
we
we'll
can
grab
search
some
around a
muscles
little
in
bit.
with the scoop,
You
right?
can stay and tell him where you want the scoops.
Okay.
You can just stay
Sounds
here.
good.
Alright, guys.
Okay. So let's suction in there, Chris, please.
Alright.
We'll try to dislodge some of the stuff, so going towards the base of the 2 worms and amongst the muscles and stuff like that.
Okay, you're on.
On jar number 1.
Yeah.
This is fine mesh.
This is fine mesh indeed, and that will be sample 108, correct?
Yes.
All good. We agree.
Yes. D'accord.
D'accord.
So that's the other end there of the suction sampler on.
It's probably at least partially.
So pretty medium. Except.
Okay. Those look great.
3 of them.
Maybe also some.
Now, 6 was okay. We want to
That was 4.
I count 10 massive steam and 2 warp steam.
Not sure how healthy the tube want to be.
Nothing as... Sorry, never mind. I didn't see that you had it on this camera.
Okay, so I think it's fine. Okay. Thank you.
All right.
We take what we need and we try not to waste.
We'll cook the rest. Candy, yeah. I'm sorry. Promise of candy. Oh, I did a little bit. Not too much.
No, it was pushed out when I put it there, so that's stuff it off.
Chris did a better job at this. Okay, let's close this big bio box.
Okay, we're good.
I'm great, Chris.
So it's not paid, mate.
What's that?
It's not paid.
Super steam.
We'll need to pick up the little man that we are bringing back to the surface.
I just make sure about this.
I've collected.
9, correct?
Yes.
Excellent.
Alright, there's this one.
Okay, so watch the stream for him. Our last goal for this dive is to collect Max's art. Thank you.
More muscle.
See it all day.
What's that?
It's not enough. Get a shot. Just those feet in the car.
Look at what's growing.
Shall we go here first?
Okay.
Okay, going down. It's all right. Makes sense.
Yeah.
It's the British
You carry the bull under your
All
arm.
right, on the pitch.
Right, okay. All right, attached.
Okay.
So good. Oh, no, not quite.
Pretty good.
A bit more. Nice.
Are you gonna keep the arm on it, or students of the animals down there as well. And come back.
And